session interaction and learning. Uh, we are going to talk about gamma irradiation, pyrogenation, and depyrogenation. We have covered an introduction to gamma irradiation in the previous sessions on the sterilization types, but here we're going to deep dive a little bit more onto gamma irradiation. Pyrogenation, depyrogenation, also we have kind of touched the basic concepts when we did the depyrogenation tunnel when we were doing the filling line concept. So we are going to do a deep dive, and I'm going to use this session to make sure that we just recap whatever was learned apart from some new uh, ideas and new information which I'll share with you. So what we are talking about in today's session is around the sterilization process using radiation, in specific gamma radiation. And the second part that we're going to talk about is the dry heat sterilization, which is nothing but heat-based sterilization, which is dry in nature. Okay, regulatory overview remains the same, that sterility is an important aspect in any sterile manufacturing process, whether it is done after the product is packaged, meaning terminally sterilized, or whether it is done at the initial stages when you are preparing the components like a depyrogenation activity, or even preparing components and getting them gamma irradiated before they come to the facility. It is a very, very important aspect and needs to be validated. Why? Because of the number of violations which were cited in the past, we are seeing that sterility remains a concern. Sterility assurance remains a concern on GMP compliance. This is just a recap of what are the different type of sterilizations, which is heat, gas, radiation, and filtration. And we are going to today talk about heat and radiation, because the rest of it we have already covered in the previous topics. Okay. So when we talk about the radiation sterilization, we have multiple type of radiations which are available. We have gamma radiation, we have electron beam radiation or beta rays, and then we have ultraviolet light, which is used for the purpose of disinfection. As I said in the past, ultraviolet light is no longer considered as a mean of sterilization. It is only used as a means of disinfecting the surface which is there, primarily because its penetration is pretty low. Then we have the electron beam, which has got, which is with the high velocity of electrons that you generate a beam, accelerated beam, that is beta particles, and those beta particles again have limited penetration, as a result of which they do not become the first choice. Whereas gamma radiation, on the other hand, is an effective radiation method only because it has got a highest penetration possible within the component being sterilized. Now, how do you generate the gamma radiation? There is, while we get into more details of how these reaction happens, cobalt 60, and I put an asterisk mark here because there is another compound, cesium 137, which can be also used to produce high energy photons, which are used in gamma radiation. So today, when we go to the industrial applications, both cobalt 60 as well as cesium 137 are widely used to generate gamma radiations. The items which can be sterilized by radiation sterilization technique are the same. Those are used for gaseous methods, which is heat labile materials, powders, plastic materials, anything which cannot withstand heat as the choice of sterilization methods. We did a sterilization decision tree some time back. On that sterilization decision tree, we had options of which kind of sterilization we can use. And so anything which is heat labile, anything which is cannot withstand steam penetrations, or cannot withstand or can get affected by gaseous method. Sometimes the compound reacts with the gases. Gamma irradiation is used. We go with a 12 log reduction in spore population. That is an overkill approach which is used in a radiation sterilization, which is the reason mainly for this is coming from the fact that when we are doing a radiation sterilization, we do not have 100% control on the beams which are going on. So instead of restricting ourselves to 10 to the power of 6, we are going to 10 to the power of 12 log reduction. Whereas from a regulatory stand perspective, we are supposed to prove at least a 10 to the power of 6. This is a typical equipment of gamma irradiation where the product is presented to the tunnel. There is a concrete tunnel in which there is a conveyor belt which moves in with the loads. So the loads are hung on the conveyor, and they keep on moving. So the load gets hung here, it goes into the conveyor, goes through the conveyor stream, and there are cobalt 60 photons which are kind of bombarded, and they penetrate the surface, and thereby they kind of sterilize the component, and they move in a path of curves and hairpin bends, so that it has got a maximum exposure within a smaller footprint area. The actual installation of this is very huge, because it's a huge concrete structure with a generator situated below this underground, and then those photons get generated, and then they get sent into the system. Now, let us see a little bit more, talk more about where gamma sterilization is used. Medical devices and drugs are most commonly sterilized using gamma irradiation. The main advantage is its high level of penetration and isothermal characteristics, which can appropriately manage heat sensitive materials, including organic metal oxides, polymer blends, or liquid crystal. 12 log reduction in the spore population. So, what are medical devices? Today, we use a lot of medical devices in our life, including a lot of wearables. We have the medical device in terms of the hearing aids which are used. We use a lot of, let's say, pens for administering drugs, like the insulin pen, or a lot of other kind of drugs. There are other subcutaneous injectable products which cannot be sterilized through the normal heat process, because they are gel-like products, and therefore they have to be sterilized. At the same time, they cannot withstand heat, so they are sterilized using gamma irradiation. We also use all our single-use components like bags, filters, hoses, etc., which are single-use and are used in the manufacturing of the sterile drug product. In such situations, also they are generally gamma sterilized. And then there are powders which cannot be heat sterilized, and therefore, in the such situation, also gamma irradiation is used. So gamma irradiation is a very widely used technique. It's a well-established technique. There is a validation around it. So that's why it is the most popular method of sterilization, though it involves usage of material like cobalt 60 and cesium 137. Very important to make sure that we justify the use of gamma irradiation, not because of the fact that it cannot be used, but also the cost of it is very high, and therefore we have to be sure that this is the only method which will do the job. Okay. Now, radiation sterilization, how does it act? We have discussed this in the past. It acts based on the damage to the nucleoprotein of the microorganism through penetration. So the photons penetrate into the nucleus, and into the cell, and then they bombard it and they damage the nucleoprotein of the microorganism, and therefore it is a good sterilization agent where it kills the microorganism. Its effectiveness, of course, depends on the dose of radiation dose or the radiation which is given to the component and the time. Okay. Gamma irradiation also ensures uniform sterilization, because what happens is that when you are putting it into a chamber like that, there is gamma, the photons are available throughout the entire surface area of the product which is being sterilized, and therefore there is a uniform sterilization method. Therefore, the risk of a microbial contamination is definitely avoided once the product is packaged. So we don't have to worry about using gamma radiation and worry about whether it is uniform or not. It is uniform. We did study in during autoclave cycle development about the worst case scenarios. Here also there will be worst case scenarios, worst case loads, but on the other side, what is very important is that it is more uniform as compared to the steam penetration and the other methods. But the cost is high, as I said, and therefore we have to take a informed decision on whether we should use a gamma radiation for a particular product or not. Okay, let's talk about a little bit of a history there. United States permitted a relatively high sterility assurance level of 10 to the power of -3 for items such as surgical drapes and gowns. However, in recognition of certain pharmacopoeia requirements as of 1998, CE marking requires SAL sterility assurance level of at least 10 to the power of -6. So 10 to the power of -3 is something that if you look at the US pharmacopoeia, earlier references, you would find that 10 to the power of -3 was sufficient to give us sterility assurance. However, other pharmacopoeias in Europe and other countries, they demanded a sterility assurance level of at least 10 to the power of -6 based on certain bio studies, and therefore it is now very important that at least 10 to the power of -6 is achieved when you are talking about gamma sterilization. Due to the number of microorganisms and drug resistance of material, the common gamma radiation dose is 25 kilogray. So this is not a default, but a minimum most commonly used is 25 kilograys, depending on the type of the material, depending on the bioflora which is present in the environment, and the component which is under sterilization, and the resistance of the material itself, because of the penetration issues. Some materials are really hard, and some powders can be very hard for it to be effectively penetrative. In such cases, you may have to vary the dose, but otherwise 25 kilograys is the standard dose which is generally used during gamma sterilization. Just a little bit of where the gamma irradiation or ionizing radiations live. We are in the visible UV visible range here, which is somewhere around 500 to 230 to 500 nanometers. Then you have the infrared, you have the microwave, which is used in the house. Then you have the radio frequency. These are all non-ionizing. They are longer wavelength and low energy. So these are where they lie. Then you have the UV and the X-ray, which is moving towards the ionizing radiation. They have a shorter wavelength as compared to the radio frequency, the microwave, and the infrared. And they do have a higher energy. A reason why UV is preferred as a method of disinfection is also because it has a shorter wavelength and there is a higher energy as compared to the visible range. X-rays are used. So these are all UV onwards are things that are used in medical profession very efficiently. UV for disinfection, X-ray for analyzing the body anatomy. We take X-rays of the body and do the scanning so that the doctors can understand this. So very effective here. And then there is the gamma ray and the cosmic rays which are there. The cosmic rays are out of our bounds, and we are only talking about the gamma ray. All of these are ionizing radiations when we start from UV onwards, and because they are shorter wavelength and high energy and do have certain amount of penetration. Okay. So gamma rays, a radioactive element emitting gamma rays or photons, that's what the gamma rays are. Photons do not have a mass or a charge. They create ionization events needed for sterilization, have a very short wavelength, are very energetic, can penetrate deep into the product, even packaged in cartons and stacked box on box, make them an excellent sterilizing agent. Okay. So why do we use gamma rays? Because they have no mass or charge, so they are easy to transmit. They create an ionization event needed for sterilization, have very short wavelengths and have a high energy and can penetrate deep because of the fact that they have a higher energy and a shorter wavelength. So it is easy for them to penetrate. So even if you had a box containing, let's say, 100 pre-filled syringes or 200 pre-filled syringes, a gamma radiation can penetrate into that. At the same time, if you use heat or any other means, it is difficult for us to penetrate deep into the surface. So gamma radiation is therefore also used when you are doing packaged compounds and you have to sterilize them, because it gives you a good penetration into the through the surface of a carton itself. Sources, as I said, there are two, cesium 137 and cobalt 60. However, cobalt 60 is the most widely used again because of two reasons. One, cobalt 60 can be produced to high specific activity. So you can generate the energy levels into cobalt 60 and control them much better as compared to cesium 137. Emitted photons penetrate deeper into the product than cesium photons. So cesium photons versus cobalt 60, cobalt 60 has a higher penetration level, and therefore it is used and also can be made into compact sources, easier and safe to work with. So it is easier to work with cobalt 60 as compared to cesium 137, and therefore the maximum literature, maximum work you will find is around cobalt 60. Now let's see how cobalt 60 works. So there is a radioactive material, cobalt 60 encapsulated. Now, cobalt 59 is an inactive natural metal. Cobalt is a metal. It's a natural metal which is inactive. There is no activity in a cobalt as a metal, which now when you bombard it with neutrons in a nuclear facility becomes partially active as cobalt 60. So cobalt 59, which is stable, plus neutrons will give you cobalt 60, which is unstable. And when cobalt 60 is left for degeneration, it will give you cobalt nickel 60 and electrons and gamma rays. The gamma rays would be in the region of 1.17 and 1.33 MeVs. Okay, so mega electron volts. Now, cobalt 60 has a half-life of 5.27 years. So if you have a cobalt 60 element and you take its half-life, it is about 5.27 years, which means that you could use it for a pretty long time without it losing its efficacy over a period of time. Sorry about that. Okay. The energy needed to create cobalt 60 is much greater than the energy of the gamma rays emitted. These gamma rays are not energetic enough to activate the product, but they can be used for other purposes like sterilization and cross-linking. So the energy which is required when we bombard it with neutrons is a very high amount of energy to create cobalt 60 itself. But when this kind of starts to degenerate and the gamma rays which come out, these gamma rays can be used for sterilization. Therefore, it is also very important to understand why this is expensive, because first of all, you have to generate a cobalt 60. Then that cobalt 60 has a half-life of 5.27 years. So if you had a kilogram of cobalt 59 and you generated a cobalt 60, you will need a lot of energy first of all to convert it into cobalt 60. And now that cobalt 60 is going to be there with you for about 5 years. After 5 years, it is no longer it has reached its half-life, and therefore the amount of gamma rays that it will emit will drop down, and therefore we have a higher cost involved in gamma sterilization. Okay. What are the major factors which affect into gamma radiation? There are three main drivers apart from the exposure time: activity and distribution within the source rack. So what is a source rack? A source rack is basically the source rack is something which is kept below where you have the cobalt 60 which is stored. So this is how a typical installation will look like completely. So you will have in the underground shipping container which contains the cobalt 60, and then you have the equipment room with the console which is generating or bombarding neutrons into it, and then the source rack. This is called as a source rack, which is where the gamma rays will get irradiated. All right, and then they are then transmitted to the upper layer where they do the radiation sterilization here. So the activity and distribution within the source rack becomes very important. The distance of the product from the source rack becomes important because you are emitting these gamma rays from here to the upward direction where the products are moving on a conveyor belt. Okay, so very important that you have this distance as low as possible. And attenuation due to product interaction, because when this now is trying to go up, and if there is a product here, then obviously that gamma rays has a shorter path. It will penetrate through that, and after that, it's no longer available. So attenuation due to shielding is also important. So we need to take care of these in the design of a gamma radiation tunnel. This is the reason why this is a specialized job, and we do not try and do it in-house. This is something which is a special arrangement which is done, requires a lot of approvals in terms of nuclear facilities or radiation facility compliances. It requires a lot of investment in terms of ensuring that the radiation does not get out, because you have the radiation area which is constructed out of concrete, as I said, and we have to ensure that though the radiation is within the chamber, it does not escape the chamber, right? And this is the reason why it is important to make sure that you evaluate a gamma radiation facility also very, very carefully to make sure that they are compliant to the requirements of the radiation norms which are laid down by in the country. There are international norms, but at the same time, there are also norms within India like BARC, which was an institute in Mumbai. The Our nuclear institutes, founded by Dr. Homi Bhabha, they have laid down guidelines for a radiation-based facility. And in such facilities, how the radiation gets generated, what should be the construction, what is the prevention mechanism of radiation getting out of the facility are all defined well. These are well defined protocols. As a pharma industry, we do not look at this sort a lot of times because we may use radiation in limited areas, but when we use radiation in radioactive labs, for example, when we use radioactivity in our labs to do our research work, these norms become very, very important, and there is a well defined protocol. There are certification programs where the people from the government of India and BARC, you have to first submit the design, then it has to be approved, then you put up the design, then you validate the design, and then you have the at and every stage, once before you start construction, after you've constructed, before you start the trials, and after you've finished started the trials, and through the life cycle of this facility, you have to make sure that there is 100% compliance, because any radiation getting out of this facility can cause damage to the surrounding areas. And therefore, it is very, very important to design the facility properly. Today, no sterile manufacturing facility will put up a gamma irradiation facility in house. Uh, Generally, these are outsourced facilities, and therefore, as a person working in a sterile manufacturing facility, if you are looking at a gamma sterilization process, then it is very important that you evaluate the vendor correctly, that they have all the measures in place, they have all the controls in place, and that they understand the sterilization requirements very, very clearly. Also, it is very important to define the size and shape of the containers which are going to go from our side as a client for gamma irradiation, and have a proper discussion so that there is no overdose and underdose. Because please remember that if a radiation is left in the outgoing container, also it is dangerous. So what is generally done is that as it moves through the conveyor, there are measures which are there to measure if there is a residual radiation which is there in the component, and if so, then it is given a slightly longer residence time so that the radiation dissipates before it comes out. So when you get a material out, there are portable devices like this which are there on the left hand side that I'm showing you, which are used to also measure if a particular container which has come out of a gamma irradiation has radiations excess of what is permissible. Okay, so these are things that are in process controls which are definitely there in a tunnel of a gamma sterilization. What is it used for? It has huge usages. It is used in medical devices. Primarily, a lot of gamma irradiation usage comes into medical device industry, pharmaceuticals, active pharmaceutical ingredients, certain APIs which need to be rendered sterile. They may be produced in a standard chemical synthesis process or in a normal process, but they, if you need to make it sterile, since it is a powder for example, and you need to make it sterile, you will send it for gamma irradiation. Pharmaceuticals, we've seen, of course, we've seen gamma irradiation of final products packaged already. We've also seen the usage of it in the components which are used in the pharmaceutical industry. Human and animal tissue, if there is contaminated tissues, then or a human tissue or an animal tissue, it can be subject to gamma irradiation to sterilize it. For example, all the implants which we get, the dental implants, the knee implants, a lot of wearables, as I said, today we are surrounded with a lot of stents, for example. Anything which cannot be heat sterilized, you would see them being gamma irradiated before they come to it. Labwares, a lot of labwares today, when you use a lot of petri dishes and others, you don't have to sterilize them. They are ready to use because they are already gamma irradiated and before they arrive at your site. Animal food and pet treats, food and spices, food packaging, cosmetics, botanicals, a huge application is there for gamma irradiation. So that is why gamma irradiation is generally something which can be outsourced also. uh, Knee replacement. So this is a knee implant. These are the various primary packaging components which are used. Human beings are surrounded by a lot of medical devices, and all of these go through gamma irradiation. Of course, the food and spice industry also uses it. Closed package products, high strength, non-breathable material packaging of raw materials. So you could do it on a closed package product. Dense materials limits permeation of steam or gas or electrons, and therefore you use gamma irradiation and unwanted chemical residues. No chemicals are involved in the process. The biggest advantage of using gamma irradiation as compared to gaseous radiations or gaseous sterilization mechanism is that it does not have any chemicals involved. So there is no residue which is left. There are no unwanted chemicals which are left. As I said, yes, there could be a element of radiation which is there, but these are well controlled processes, so we will not see a residual radiation coming out from a gamma sterilized product. But at the same time, there are no chemical residues or chemical interactions, which is why it is a method of choice when you can't use steam or heat as a method of sterilization. Always go by the sterilization decision tree, eliminate the thing, as we said. The first choice is, can you terminally sterilize something? If you can terminally sterilize something, then go, do not choose a aseptic process. If you have to use an aseptic process, then you have to worry about components which are used in aseptic process, and therefore you may have to use some of them through steam sterilization, some of them through depyrogenation or heat based sterilization, and then of course, if there is no other choice, then gamma irradiation. As I said, you could continue to use ethylene oxide or VHP as one of the other methodologies, but in either cases of ethylene oxide or VHP, there is a chemical residue fear, and therefore, if possible, you will gamma irradiate those components. Today, when we talk about aseptic manufacturing or sterile product manufacturing, any ready to use primary components like syringes, for example, they come in a nested format. I have probably talked about it when we discussed the filling lines. We use a lot of single use products like fermenters to buffer tanks to hoses to a lot of other single use components in the industry, filters for example, and they need to be sterile before they are used, and in such cases, gamma irradiation becomes a choice of method of sterilization. So the choice is always based on if you can do it in-house and terminally, that's the first choice. Then you move to aseptic processes, and then of course you use ready-to-use components and single-use where applicable, where gamma irradiated components would be there. One of the processes that currently I'm running is a process where we are manufacturing an injectable product which is delivered subcutaneously, which means it goes deep into the skin before it can be administered. It is used for cancer treatment. In such product, it's a very high viscous product, and we cannot sterilize. We can filter only one part, which is the water. We may be able to also sterilize the powder before it is mixed, but the whole process of creating the gel cannot be made into an aseptic process. It's very difficult because it just converts into a gel like Vaseline gel or any kind of a gel that you use. It's like a viscosity of 150,000 CPS. In such situation, after the product is formulated and it is filled into syringes, we send the entire syringe for gamma irradiation and make it compliant to the sterility assurance levels required, which is 10 to the power of -6 minimum. Of course, it's with a 12D overkill approach, so 10 to the power of 12 is what is monitored. But and this product is a very expensive product from a treatment perspective, and therefore it is very important that we have a gamma irradiation, effective gamma irradiation of the product, so that we can have this. And in a gel, you can imagine that if you took, let's say, Vicks VapoRub, right, which everybody uses at home, how difficult it is to steam sterilize it, or how difficult it is to heat sterilize it. If you try to do a heat sterilization or a steam sterilization of that, what would happen? It would form an emulsion, first of all, because it's a jelly material. Second thing is, it would just disintegrate. The products would just get destroyed because of the heat. Now, if you had to sterilize that, and if you use a VHP or an ETO as a gas, is it possible to penetrate that thick layer of Vicks Vapora? Very difficult. But if you wanted to make that Vicks Vapora sterile, you don't need to make Vicks Vapora sterile. But if you wanted to make it, then gamma irradiation would be what you would choose, because it can penetrate through that high viscous product. When bombarded from all sides, you will get a sterile gel out of that. So that is the reason why gamma irradiation has to be chosen carefully when used for pharmaceuticals. So with this, we come to the end of the gamma sterilization as a topic. I'm going to jump into depyrogenation because we have touched upon it already earlier. So I'm going to run through that, and of course, then take the question and answer on both the sessions. Depyrogenation. What is a pyrogen? It's a molecule or a substance that causes a fever, also known as pyrexia. Most common type of pyrogen affecting human is endotoxin, and most commonly the bacterial endotoxin. So we have already discussed this in the past. This is a slide from the previous sessions. I brought it here as a revision slide so that you can go through that. I'm not going to spend too much time on it. The maximum acceptable level of endotoxin levels is 5 EU per kg body weight. 100 picograms, sorry, 1 EU is approximately equal to 100 picograms of E. coli liposaccharide. And what is permissible is when you are exposed to 5 EU per kg body weight, you can develop symptoms of fever itself. So the US FDA has limits which are set. Injectable drugs which are intrathecal can be zero point 0.2 EU per kg body weight. If it's a non-intrathecal, it has 5 EUK per kg body weight, and if it's sterile water, you have 0.25 to zero point 0.5 EU per ml, depending on the intended use. So these limits are fixed. So endotoxin is a subject of importance, and to eliminate the toxins or endotoxins, we use the process called as pyrogenation or depyrogenation. Okay, so it is a process which is used to remove the pyrogens from a solution or a component or a surface using hot air at high temperatures. Depyrogenation is crucial to the safety and efficacy of pharmaceutical products, because if the pyrogens are present in the product or the compound which is delivered to a patient, then they can develop symptoms which are negative and can lead to complications on an already existing diseases. So it is very important that there are no endotoxins in the product which is being administered to a patient, and therefore there are clear limits which are defined for all the products which go out of a sterile manufacturing facility. Okay, we have seen how a depyrogenation tunnel works, so I'm not going to spend time on it, but I'm going to spend some time on this discussion. Again, D value, Z value, like we did for the moist heat sterilization, for depyrogenation also, D and Z value becomes important. Here you will calculate the D value, the Z value the same way that you calculated for it. The only difference here is that for here, instead of an FO, we call this as an FH value, because this is based on a heat value, not a F0 value, where you are just making a difference of log reduction in the microbial load. Here you are using dry heat sterilization to eliminate the pyrogens, and therefore you call this as an FH value. Okay, formula remains the same. 170 is used as the baseline here instead of 121, whereas in dry heat sterilization 170 for a sterilization time for a certain period is more than sufficient, and therefore we use heat penetration factor here also. The calculations will remain the same as what we did as an exercise during the moist heat calculations of FO values. We can calculate T or the time, we can calculate the temperature, or we can calculate the FH value. So all three of them can be calculated using this formula. Okay. Uh, where is dry heat sterilization primarily used? It is used in items that are damaged by moisture, or for material that maintain their structural integrity when subjected to elevated temperature without undergoing melting, combustion, or alteration. Powders are one of them. Petroleum, some of the petroleum products, sharp instruments can be sterilized using dry heats. Also, all your glassware which is used in the pharmaceutical industry for filling of the pharmaceutical injectable products is sterilized using dry heat only. Sterilization uses the principle of conduction. The heat is absorbed on the outside surface of the item and then passes to the center of the item layer by layer, and will reach the temperature sterilization to take place. Okay. How does it work? It oxidizes the molecules, or causes an irreversible denaturation, and therefore it destroys the organism or the cell constituents, by virtue of which the organism dies. Uh, we have looked at depyrogenation tunnels where the temperature go as high as up to 300 degrees centigrade for a short period of 3 minutes, because there you are looking at a passage of a vial, or a cartridge or a PFS into a tunnel which is on a continuous feed basis. And if you had a very high speed filling line like a 200 PFS per minute or a 600 vials per minute, then obviously what is important is that you need to go at a higher temperature in a shorter time. Okay. uh, Bacillus atrophius is used as a score for monitoring the bacterial or the biological indicator, which is used for measuring the dry heat sterilization efficiency. Okay. This again is a curve which we have seen in the past, where what are the different phases that you have the preheating first, then it goes to the temperature at which you have to hold it, then you hold it. You do a final heating where it reaches the actual temperature 270, 250, whatever the temperature is. Your hold time begins, and then you start to cool it, and then you remove it from the system. So this is a batch process which is there. In case of a tunnel, what happens is that when you are feeding it from a washer, you preheat it with the exhaust air, so the temperature of the component starts to already reach a higher value, and then you increase the temperature all the way to 250, 270, 300 degrees centigrade. After you reach that, you hold it for a minimalistic time what is required, and then you subject it to cooling, and then you take it out and subject it to fill finish operations. So a lot of materials can be dry heat sterilized, and as long as they can withstand the heat. So with this, on depyrogenation, since we had already done in the previous class and covered a lot, there are no new factors except the FH value, which is more elaborately expressed here. Of course, that formula was also available in the previous slides. So I will now open the session for question and answer on these two topics of gamma sterilization and depyrogenation, and then we will move in the next session after a 15-minute break to talk about how do we validate the gamma sterilization, and also the concept of gaseous sterilization validation. So we will talk a little bit about the validations here. That's the next topic which we are going to cover. And then we will move into the last session today, which is introduction to biopharmaceutical manufacturing. So now I throw the session open to question and answers by students. Please feel free to ask questions and clarify your doubts. And I'll be happy to go back to any of the slides that you need me to go back to. So, any questions?
Sir, I have one doubt.
Yes, yes, Shankar.
Sir, non-endotoxic materials also can cause the pyrexia, sir.
And no, endotoxin is the major reason for causing of pyrexia. Now, what does that mean? What is an endotoxin? An endotoxin is a dead material. The cell, after it is, so you remember the microbial death kinetics that we covered in one of the sessions, right? So what happens is that you have a growth medium, the cell starts to grow, it starts to reproduce, and then it starts to naturally die, just like any life on this earth has a limited time and it starts to die. Even the cells start to die. When they start to die, when they start to get into the death phase, then they start to emit out or toxic materials from their body, which is what is the endotoxin. Now, endotoxin, why does it cause pyrexia or create? Because it produces a pyrogen. A pyrogen is nothing but a toxic substance which enters into your body, reacts with your body cells, and therefore tries to create a situation where you will get into a pyrexia or a feverish situation, because your cells are trying to fight a toxin. And if they are not able to fight a toxin effectively, then they will react in the body, creating a high temperature rise in the body itself. You see, a body is a very beautiful machine. A body has its own inbuilt mechanism to fight everything that we face today. Any external contaminant which enters into the body, we have the cells which are healthy cells which try to fight them out. And the first symptom of any fight that your body is doing is a rise in body temperature. Always remember this, that rise in body temperature means that your body is trying to fight something, and therefore you have a contamination inside your body which needs to be cleaned up. Okay. There are, of course, you can take medicines to do it, but there are certain natural ways also of doing it. But if the level of toxins go higher, if they go more than 5 EU per kg body weight, then obviously they can get into dangerously high situation. So sometimes we see people dying of high fever because the endotoxin levels in their body has gone up very, very high. So the answer to your question is that there are no non-endotoxin materials which can cause pyrexia. That's a specific case, but you will still see fever coming into your body when a body is trying to fight something. Okay. Today, if you let's say go out and have something which is not right, some food which is contaminated food, contains some, the first symptom you will see in your body is what? Fever. Why is that fever caused? Is because that particular contaminant is causing some death in your good cells, which are releasing toxins, which is increasing your temperature. But the body has a natural tendency to fight it and will fight it if your cells are good. But if your cells are highly contaminated, then that fever starts to go up, needing you to take an antibiotic to kill the contaminant within your body. Clear?
Yes, sir, clear.
Okay. Okay. Anybody else has any questions? Okay, so if there are no questions, let's kind of take a break here. We will rejoin at 10 o'clock, which is another 12 minutes. And we will get into the session, which is around the validation of gamma radiation and gaseous based radiation sterilizations. Um, so let's meet sharp at 10 o'clock. Yeah.
Okay, sir.
All right. Thank you so much.
Thank you, sir.
Thank you, sir.
Okay, so is everybody in? Yeah, looks like.
No,
So
sir.
we'll start.
No, sir.
Not yet?
No, sir. I'll
Okay,
just.
okay, fine.
Sir?
Can we start?
Yes, sir.
Okay. So welcome back everybody to another session. Uh, this session we are going to cover validation of gamma radiation. We spoke about gamma radiation. We spoke about gaseous and radiation-based sterilization. Now, how do we validate these gamma radiation? What is involved when we decide when to do gamma radiation, how to do gamma radiation? So again, we are going to cover the chemical part of sterilization and radiation part of it. And primarily, we are going to talk more about hydrogen peroxide because that's what is most widely used today, and gamma radiation as the two processes which will go for validation. When we talk about gamma radiation validation, we first talk about what are the guidelines which are available. Okay, the validation before we start the routine gamma radiation process. Okay, a product with a sterile claim need to complete a validation process to ensure that the sterility assurance levels which we are claiming are being met. Now, there is a guideline which is ISO 1137-2006, which provides the guidance for completing such validation through three parts that are included under the general title Sterilization of Healthcare Production by Radiation. Okay, so this is an ISO guideline which is there. It has three parts. Part one, which is titled as Requirement for Development, Validation, and Routine Control of a Sterilization Process for Medical Devices. Part two, which is establishing the sterilization laws, and part three is the guidance on dosimetric aspects. So let's just understand what this is. Part one of the standard covers the requirement for developing, validating, and controlling the radiation sterilization process. So, how do we first of all control the radiation process, the sterilization process? How do we validate the process? This is what is covered in part one. It talks about both cobalt 60 and cesium 137. So, from a perspective of a guideline, it covers both the molecules. So, cobalt 60 is what we will use more often. Part two is the information about the method manufacturers will use to determine the minimum necessary dose to achieve stability. So we spoke about 25 kilograms as the standard use, right? But it could be 25 kilograms, it could be 15, it could be even 40. So how do you decide this dose? This is what is covered in part two. And a kilogram is a unit used to measure the gamma rays that are emitted as the radionuclide decay. So when decay occurs and we saw that the gamma rays are emitted, so that's a unit which is used to determine the decay or the emission of the gamma rays as the radionuclide is decay. So that's why the unit is kilograms of the time. Part 3 of the standard offers guidance for manufacturers to meet the requirement of part 1 that relate to dosimetry, which is the measurement of the gamma ray dosage and its role in developing, validating, and controlling the sterilization process. So this is more for the gamma ray dosage measurement, which could be either at the edge, at the end where the gamma irradiation process is done in the tunnel, or when you have the product coming out and you want to measure what was the gamma ray which it was subjected to, then and what is the residual radiation, then part 3 covers the guidelines for this. So the ISO 1137 1137 2006 is what is followed for gamma irradiation validation. Now, when we look at the gamma irradiation validation, there are various steps which are described through these three parts, and we're just going to cover some of these stages so that you understand what is involved in the whole process before we decide that a gamma irradiation is the choice that we want to make. How do we what are the process steps involved? You have decided you want to do gamma irradiation, but if you want to do gamma irradiation, you need to follow certain steps. So these are the steps which will be where you can validate the process more efficiently. First is the product design stage. When designing the products to be treated for sterilization, consideration should be given to the material challenges on the process of the process on the product and packaging. What does this mean? What it means is that when you are designing the product, when you are designing, it could be, remember we talked about polymers, we talked about medical devices, we talked about product filled into these primary packaging containers like PFS or so. In such a situation, we have to consider what could be the challenges that could be there on the process of gamma irradiation because of the product and the packaging. The gamma irradiation process can sometimes modify the polymers, though it's a non-reactive. It can add an ozone odor. It can decolor sometimes the product, or it can also sometimes modify the polymer slightly because what are you doing is you are bombarding it with a radiation or with energy. When you impart energy to any polymer, obviously there could be some degradation of the polymer if the radiation level is sufficiently large to cause that kind of a decay of the material itself. So it is very important to first of all consider what the gamma irradiate could affect onto your packaging material, and therefore what packaging material one should use. So it's very important to decide on this, saying that, okay, if I'm using a certain kind of plastics, and most of the times we are using a lot of plastic containers, whether it is talking about single-use bags, whether it is talking about single-use hoses, whether we are talking about those eye drop bottles, whatever we are talking about, or even if we are talking about PFS, where the product is filled, they could be made of glass or they could be made out of polymeric material. Polymer is a large choice of material. All your medical devices, your knee implants, your hearing aids, your a lot of other products, including stents, are made out of polymers nowadays. So when you are using a polymer and you want to gamma irradiate it, you have to have a good knowledge of polymer chemistry to understand that when you apply gamma irradiation, what could be the probable effects, and therefore the dosage can be decided based on this aspect also, or very, very important factor to consider. The other consideration is that a series of product cartons will be loaded into an aluminum container called a tote or a carrier, which will convey the product around the radiation source. So you saw that conveyor in the picture where the load was moving through zigzag or a hairpin curve path. In such a situation, you are going to load your cartons or whatever your products are into an aluminum container. It is going to be having a metal container in which you will load the products, and then you will bombard it with the rays. So standardizing the product carton sizes and densities in an effective manner will offer the most economical return on the irradiation process. So very, very important to understand what should be the size of my carton. If I make it too large, or if I make it too non-standard as a size, then I probably will end up using different kind of an aluminum container or a specific aluminum container which may be true or usable only for me. However, if you know what kind of aluminum skeleton is being used by the vendor for placing the cartons before they do the radiation process, and you decide the size of the carton and how many products can you load within that carton of yours, which is then subjected to gamma radiation, it optimizes the overall process so that it gives you a good economical way of doing gamma radiation. So very important to make sure that we understand the product design very well. One is the product itself, polymer product, how does it get affected by gamma radiation? Second is how do you package it before it goes for gamma radiation so that you can have effective radiation process at a low cost, at a reasonable cost. I'll say low cost because if you did a non-standard packaging, then the cost of doing the same gamma radiation would be high. Okay. Now, bioburden determination. This is very important. Now, the typical gamma sterilization process is unique in that the manufacturer uses natural microbial load present on the product and sealed packaging to determine the processing parameters. So, generally, statistical representative samples of the product will be submitted to a microbiology laboratory to quantize the population of colony-forming units through bioburden determination. So, unlike our bio indicators, which we used in moist heat sterilization, like we used in dry heat sterilization, we even used in vapor hydrogen peroxide. Here, the usage of bioindicators is difficult. We could use bioindicators here as well, but what is generally done is that you take the bioburden count of the load through a representative sample of the product which is being subject to gamma radiation, and then you decide the level of the dosage. So there is a dose optimization which happens based on the expected load and the microflora which is present. Uh, so this is very important that we have a clear idea of what kind of microbial load you have, characterize the microbiology flora which is there, the microorganism flora which is there, so that we know that once the gamma irradiation is done, whether we have achieved an effective kill or not. So that's important here. Verification and material testing dose determination. Now, based on the bioburden results, there are tables which are provided in ANSI ISO 11137 require, which will say that okay, you to achieve a specified sterility assurance level of 10 to the power of minus 6 or 10 to the power of minus 12, this is the amount of kilograms that you have to use. So this is based on certain studies which are done globally, and before the ISO 11137 2006 guidelines were published, and those tables are available. These tables, of course, get updated over a period of time, and those updated tables will also be available today over a period of time. And based on this, general guidance is provided saying that okay, you can use 25 kilograms, or you can use 15 kilograms, or you need to use a higher number of kilograms so that you achieve the required sterility dose. So first is to determine the dose. Now, multiple methods are available for determining sterility within the guidelines based on the level of sterility assurance, selected processing dose, and the number of samples which can be used for tensing. So there are methods which are provided saying how do you determine whether the product has been become sterile in the ISO 11137 guidelines. Okay. It is also recommended that a high dose material test be performed by irradiating products at least 1.6 to 2 times the expected minimum sterilization dose, or 50 to 40 to 50 kilograms if the minimum dose is unknown, to evaluate the attributes of the product after processing. So you do also a high radiation dose test, meaning that remember we spoke about 25 being the standard dose rate, and this comes from the ISO 11137 guidelines. So you also do a high dose material test, saying that if what happens if I expose it to 1.6 to 2 times that dose. Now, if that dose is not known, if for any reason the table does not provide you the dose rate, then you assume 25 kilograms as the base dose, and then for the high dose material test you use 40 to 50 kilograms because you don't know the minimum dose rate, so that you can know what is the effect if you expose it to higher dose. So it's like a bracketing approach where you are doing by saying that I will not go above this, I will not go below this, this is where my optimum study is. Verification and material testing. So now what happens is that once you have a verification dose in the ANSI, when you look it up, the verification dose is generally very low and is required to be within plus 10% of the verification dose reference values. This type of irradiation is very difficult to achieve in a large processing irradiator, and typically requires a small research irradiator to achieve the ISO requirements. The research irradiator will apply the required verification dose by monitoring the imparted dose by placing quantitative measuring devices on the product package called dosimeters. If the material testing is also desired, it can be submitted at the time of irradiation. So let's understand this a little bit. Now, if you're when you subject a product into a large irradiator, the tunnel which we saw, then to control the dose at very low levels within that on such a large equipment becomes sometimes difficult. And if you wanted to determine that without, and I have a small, when you start a product development of gamma irradiation, you're not going to have thousands of packages. You're going to have 10, 15, 12, or 20, 25 packages which you want to subject to gamma irradiation. So what do? You take it to a lab irradiator, a small research irradiator, and then you subject it to low dose rates of whatever has come out from the table, and then use dosimeters to find out what is the actual level of dose which you have imparted to the product. And based on this, you can determine whether you have effectively achieved your sterilization levels or not. So you have to conduct these small scale studies before you subject the larger material to irradiation itself. Of course, the material visibly may not kind of look decayed or decolored to the natural eye. So there could be material testing which you want to do. You take a non-radiated material, send it to a lab for physico-chemical parameters, including microscopic evaluation of the structure. And then after it is conducted or subjected to gamma irradiation, you send that same irradiated material to the same laboratory and evaluate it on the same parameters to say that because of the gamma irradiation, your packaging has not deteriorated drastically. It may deteriorate slightly and may be stable. So again, stability studies will also have to get done over a period of time if there is a deterioration. But you do a material testing as well. So you have to do verification in material testing experiments once you decide the dose levels. Sterility testing. Once the verification dose has been applied, the product samples will be routed back to the microbiology lab to perform the sterility test. The sterility test will confirm whether all viable microbes present on the product have achieved lethality to deem the process successful. So this is a no-brainer that you are doing the chemical analysis. The second one that you're doing is to ensure that the dosage is right, and the third one is primarily the sterility testing, because the whole purpose of doing gamma irradiation is to achieve sterility. Now, once you have done this verification, etc., then you decide what should be the sterilization dose based on the sterility assurance level. So, when we go through the steps, you first do the product, you study the product. This is all polymer chemistry and designing the product right. How you're going to package it, how you're going to put it there, what is the initial bioburden which is expected in the material in a normal condition. Then you look at the data on the 1137 11137 guidelines to make sure that you understand what should be the dose which is there. You also use a irradiation technique to determine what is 1.6 to 2 times. You do this verification at a small research irradiator, do the study on the materials, and then also do the study on the sterility testing, based on which all the data that you've done, and this is just not one experiment, you have to do multiple experiment, you have to do repeatability there as per ISO guidelines, saying that you will subject the say under the same condition at least 3, 4, or 5 times the same dosage rate, and do a verification of the material, verification of the dosage, verification of the sterility, so that you are 100% sure that the dose that you are determining will do its job of sterility assurance that you're looking at. Okay. Now, during the routine irradiation, the product volume and density will increase as it is processed in a large carrier or tote. Dose window or a range needs to be established for a large irradiator to process the product effectively. The chosen sterilization dose range will be the minimum dose needed to achieve the sterility claim, and a dose level value below the dose applied for material test as the maximum end of the range, or generally a minimum of at least 1.6 times the sterilization dose. A dose level value below the dose applied during the material test is recommended as maximum end of the range, or generally a minimum of at least 1.6 to 2 times of the sterilization dose. So, what does this mean? Uh, let us uh, let us understand this a little bit. Now, when you are going to go into a large irradiator like the plant scale irradiator, there are other factors which come into play. What you've done so far is at a lab scale in a controlled environment in a very limited space. Now, when you're trying to do this at a large scale, then you cannot just stick to the minimum dose, because if you stick to the minimum dose, there could be certain shadow areas within the irradiator, and therefore your product might not get sterilized. So you define a range or a window saying that I should not be below so and so dose rate and should not be higher than a certain dose rate, because both doing it higher than a certain dose rate and doing it lower than a certain dose rate will have a contradicting or a detrimental effect on the product. If you do a low dosage, you will not achieve sterility. If you do it higher, then you might deteriorate the product, or you might decrease the shelf life of the product, or both. In such a situation, you have to fix a window. So let's say you arrived at 15 kilograys or 20 kilograys as the value at which you are supposed to dose. You will fix a dose rate which will be something like, let's say, 22 kilograys to 27 kilograys, because what is the 1.6 times the minimum dose rate of 20 is 32. So you want to be staying below 32 and within higher than the minimum dose rate of 20 that you've achieved. So you will define saying 22, 23 to this. How do you determine this? You determine this by fixing the range from a minimum dose rate at which you achieved lethality, and then taking an assumption saying that cannot go below 15, I cannot go below let's say 20. Now therefore I want to keep at least something higher than that, and that higher is at least 20% higher or something like that. Again, ISO 11137 gives some light onto this of how to determine this range also. But what is important to remember is that we need to fix a window, because in no irradiator of a commercial scale you will exactly see the precise dose of 15 or 20 all the time. There will be a variation, because remember the construction of it, remember the storage rack, the rack that we spoke about in the earlier presentation. So based on this, you have to fix a window and work it with a window. Now you will ask me a question, why take a chance? Why can't I just go to the higher limit and fix the range? Again, if you fix a range at a higher level, and if any value falls below that when measured in a manufacturing process, or the tunnel actually, when it moves through the tunnel, through those hairpin bends, if at any given time your dose rate falls below the specified value also, you cannot accept the material as sterile. So that is the reason you always provide a range, just like we did in temperature. You always say 123 plus or minus 2. You never say 121 degrees centigrade. Why? Because there are variations. Your steam pressure will vary, your moisture in the steam could vary, your temperature would fall because of a certain valve opening out and allowing the condensate to go out. In a similar region, you do not want to go below 121 anytime. So what do? You fix a range and say, I will do my sterilization at 123 degrees centigrade, and I will not allow it to fall below 121 degrees centigrade on the lower side, and on the higher side I will not go 125, because I have a possibility of charring the material. In a similar way, here in gamma irradiation also, you will fix a window of a dosage saying that I will not go below this, and that below is generally slightly higher than the minimum dose range, and I will not go above a certain value, which is definitely not more than 1.6 times the minimum dosage rate, so that you are able to make sure that you always achieve your sterility assurance. Okay. Routine processing facility determination. Once the sterilization dose range is established, there are technologies which are there. Steris has one very well established sterilization technology. They will tell you what is the best processing facility to routinely process your marketable products. Okay, so you have to interact here with the manufacturer of these gamma irradiators to make sure that you understand what is the range at which what is the right kind of a tunnel in which, or the irradiator in which you are going to do the gamma irradiation. So you need to have that definitely determined and discussed with the service provider. The processing facility will ask that processing specification and new customer setup documentation are completed to provide the means to identify all incoming products and required dose range to achieve the sterility claim. So you have to then make up a documentation, so that now you're introducing a new product. you have a processing specification which are determined through these dose rates. So entire documentation has to be provided saying that this is my incoming material, this is the dose range which I want you to do, and this is the sterility claim that I'm making on this product. And based on this, you are going to make sure that this documentation is handed over to the processor. Once the product is received by the processing facilities, measurement of the product carton and density will be made. The facility will establish if the like product density and volume has been processed and will perform a dose mapping of the product if required. Dose mapping is performed by determining the most efficient means of placing a product in a carrier or a tote and placing numerous dosimeters throughout the product load to establish the minimum and maximum areas of imparted dose. Once these areas have been determined, routine dosimeter placement will be limited to the minimum and maximum dose zones in a select sampling of a routine processing torch or carrier. So once you have now given the documentation, you have the data, you have the window in which you are going to do the dosages. Now what next? When you go to a processing facility, if they already have done a similar load pattern or number of product cartons with the density. So here it goes by the volume, the size of the carton converted into a volume and weight. They will, if they have already done that kind of a process earlier, then they would probably know where to place the dosimeters during the process, the routine process itself. However, if they do not have that kind of a data, and while your tote size or whatever you are used is fitting within their carrier, but your carton size and volume and density are different from what they have processed in the past, they will perform what is called as a dose mapping. Now, this is very similar to our biological indicator placement on a full load at different places to determine whether you have achieved the right doses and what is the maximum dose that you have achieved in that load area, what is the minimum dose which you have achieved. Similar to also thermocouples placed within an autoclave to make sure that you study the temperature at various points or zones or within the loads of autoclave and then determine which is your controlling factor. So during a routine process also you have to monitor it. So how is this done? This is done by identifying what is called as a cold spot in a temperature-based sterilization. In this case, it would be minimum dose and the maximum dose. So you will determine the hottest spot and the coldest spot, or the minimum dose spot and the maximum dose spot. During the routine operation, you will place dosimeters only in some of these locations. There could be more than one minimum spots or more than one maximum area doses. So you will place dosimeters only limited number of dosimeters during routine process. But during the mapping, you will place it throughout the load and make sure that you identify the penetration correctly. So this is an important exercise. So you first design the product, then you design the packaging, then you determine what should be the volume, then you determine the dose rates based on theoretical data. Then you conducted a small irradiator study. You fixed the range. You then decided to give this to right kind of a facility. Now you did the documentation which was required. Now you are doing the mapping. So this is the process of validation which is happening. Routine processing, once the processing specifications are established, routine processing irradiation will be identified by the product code as received at the facility, and irradiation process will commence without further requirements from the customer. Although it is recommended that if any product changes, size or density or priority, turn time requirement occur, the customer contact the processing facility for communicating the unique requirement. So what happens is that once these are established, then it becomes a run-of-a-mill process for the irradiator fellow. And he will send your totes because you may not have sufficient loads to fill his entire tunnel. Now, let's say your dosage rate is falling at 25 kilogray plus or minus 5 kilogray. Okay. Now, in such a routine processing, when he is doing 25 kilogray plus or minus 5 kilogray, he will take your load along with other loads which are in the similar range They will be identified using a product code. So you will package it using a product code. He will put it inside the irradiator, which has got other products also, but they are all running on the same dosage rates, and he will subject it to gamma irradiation. If there is any change on the size, density, or any aspect which you are sending to the processor, it is your responsibility to contact the processing facility and say, I have changed this. Whether he can use the same parameters or something has to change, has to be advised to. Okay, so this is a very important coordination activity, making sure that every time you send a product, it is packaged the same way, the same size, same density once it is established. If there is a change, there has to be proper documented communication, and there probably are more explanations to be given to regulator why something changed. Maybe your source material changed, your carton was being produced by one approved vendor, and that vendor went underground for whatever reason. You had to qualify another vendor and get the carton boxes from them. There was a slight variation in the size of the packaging, the way you filled the product into that carton. So if that kind of a small change also occurs, you have to inform the processing facilities. Based on your dosage studies, you might still determine and say, I want to use the same sizes, but irrespective of that, you will go ahead and inform if there is any change in the size of the density. So this is very important, which are there. Those audits as initial sterilization dose determination was based on natural bioburden of the product packaging line. The 11137 standard recognizes that microbial population can change with seasonal or periodic manufacturing changes. Thus, the standard requires a bioburden determination and dose audit at a verification dose level to confirm the minimum sterilization dose being still valid. These irradiations would be performed at a research irradiator at the same test constraints of a verification dose, in addition to performing a sterility test to validate effectiveness. So, what does this mean? That the bioburden or the flora, remember, we are not using a biological indicator here. We are dependent on a natural bioburden. Now, the natural bioburden will change. Bangalore had a beautiful climate 30 years back when I came, 32 years back when I came here, or 35 years when I came back here. At that time, the microflora was totally different. Today, we live from a garden city into a concrete city. In that concrete city, obviously, the kind of bioburden has changed. The people who used to know old Bangalore know that there used to be a lot of pollens which were there because it was having a lot of flowering trees, and people having asthma had issues living in Bangalore. Whereas the people who did not have that issue loved Bangalore for its weather and all. Today, the same people who used to have asthma issues can easily live in Bangalore because the number of pollens have fallen down. On the other side, people who have lived in the earlier Bangalore find it too hot to live into that. So these bioburden changes will happen. We have seen the the advent of COVID-19 kind of viruses into the environment. Obviously, our microflora is changing with every season over a period of time, and therefore it is important to make sure that the natural bioburden tests are done on the product and the packaging routinely at a certain frequency to make sure that we are still achieving the right sterility assurance levels. So this is called as a dose audit. So this with this I come to the end of gamma irradiation validation. So gamma irradiation validation involves a proper documented procedure of right from the design of the product to packaging to dose ranges to doing the verification at the manufacturer site, and then doing a routine analysis plus audits over a period of time. And this is how you validate a gamma irradiation process. So, as you can see, it is not very simple. Biological indicators you would fall in love with now because they were so much easier to deal with, right? Here now you have to do a lot more work to get a sterility assurance level done. And if the microflora changes, and as a result of which your dose does not work, then you have even a larger problem. So with this, you can see that how much the biological indicators have given us a beautiful support in terms of the sterilization validation process, which in gamma radiation is not possible. So with this words, I come to the end of validation of gamma radiation. Let's get into the gaseous sterilization validation. Sterilization can be accomplished using various gases like hydrogen peroxide, peracetic acid, formaldehyde, ozone, etc. This is what is written in USP 43. So USP 43 recognizes these as sporicidal agents which are suspended in air and sterilize them being used for sterilization. You can see that certain things like ethylene oxide and things do not now find a place here because of the fact that they are carcinogens. So only the safer chemicals have been listed here. Uh, Okay. Now, gaseous sterilization procedures are the exposure of items to a chemical sterilant or in a gaseous phase in a controlled manner to ensure that a predetermined lethal effect is delivered to the items being processed with the objective of eliminating pre-treatment bioburden and providing satisfactory margin of safety. Again, gaseous sterilization process definition here, it is emphasized that end product testing does not guarantee a consistent or high quality product, can only effectively identify and statistically quantify the incidence of substandard product. The gaseous sterilization procedure in conjunction with the remainder of the manufacturing process must be designed to maximize the probability of product being suitable for intended use. Now let's understand this statement, which is again coming from the regulatory stand perspective, that if you just do a sample of the finished product which is subjected to gaseous sterilization and you find that it is meeting the sterilization sterility assurance, it does not guarantee that that is consistent or that is true for the entire batch, right? Because they could be, you cannot sample the whole batch, otherwise your whole batch will become non-usable, because sterility testing is a destructive form of testing. So you will draw only samples, but what you have to do that when you are doing a gas sterilization process, you have to ensure that the upstream and the downstream of these process are also validated time and again, so that you are 100% sure that you have not introduced any contamination even by mistake. You will still do a sample based analysis only, because you can't do a full batch analysis of microbial counts, but you have your friend biological indicator which is already available, which will tell you that your process of sterilization was effective. So you will use a combination of biological indicators, sample-based system, and the analysis of the batch upstream and downstream of the gaseous sterilization to make sure that the product is suitable for intended use. Okay. What needs to be done before you start a gaseous sterilization program? You need to do a product definition in terms of physical, chemical, microbiological, and pharmacological properties where appropriate. So again, similar to the validation process that we look for gamma irradiation, you need to understand your product and make sure that it is compatible with the gas that you're going to use from a physical chemical property. You should know the microbial flora which you are trying to get rid of, and the pharmacological properties that by usage of the gas, because remember that hydrogen peroxide, peracetic acid, formaldehyde, ozone, etc. are all oxidation agents, and when they oxidize, if for any reason the pharmacological properties of your drug product or a drug substance is changing because of the sterilization process, then that needs to be properly studied. Okay. Specification of the raw materials and the components is important. Again, raw material here would be what is the percentage of hydrogen peroxide and what quality of hydrogen peroxide you're using. Components would be, again, if you are using any kind of a packaging around it, we use Tyvek bags, we use sometimes plastic containers, which are there, open containers through which you will do the VHP circulation. You need to have those specifications defined. Determination of the required sterility assurance level based on the use of items being treated. So you will put the actual items for treatment and do a swab test or a biological indicator placement, and then determine whether your sterility assurance levels are achieved. Compatibility of the process with the items being treated, very similar to point number one, that it should be compatible. Determination of the acceptable limits of the major residues after gaseous sterilization procedures. Remember, when we did a study on ethylene oxide, we talked about residues left by ethylene oxide and the limits which were defined in the pharmacopoeias for these residues. So it is very important to understand that the residues are not above the acceptable limits. And of course, the validation of analytical methods used with adequate calibration and qualification of the measuring equipment. Now, you are using various analytical methods to determine the acceptable limits, the physical properties, the chemical properties, the pharmacological properties. You're also using instruments to determine the microbial loads before and after the sterilization process. So whatever methods you are using for quality control or quality testing have to be validated themselves to make sure that there is a proper measurement being done, and because of a fault in the analytical method you are not getting a false result. Okay, so very important that in pharmaceutical processing, in sterile manufacturing process, or any other process for that matter, we have to always have all of our processes validated, meaning including your analytical methods. Now, when you validate the analytical methods, you use chemicals in those methods. Those have to be also qualified. So qualification of components, equipment, systems, processes, training of people, all are very important when you conduct a pharmaceutical process. We will cover in a session tomorrow on the aseptic behaviors, right? And one of the most important aspect there is how personnel can cause contamination, and therefore it is very important that we ensure that there is a validated process and there are methods, analytical methods, to find out any deviation from such a result. Okay. In some circumstances, a satisfactory demonstration that a gaseous sterilization procedure consistently produces the desired sterility may eliminate the need to test every batch of items being treated for sterility. So, if you are able to consistently reproduce the results of sterilization or conduct the sterilization process repeatedly, then you are 100% sure that your sterility assurance is being achieved, and therefore you need not test every batch. So this is called as a forward release method, where you kind of release a batch even without a complete testing because you are 100% sure about your validated processes. You can release batches. This is permissible under the regulations, provided that you have properly documented every procedures, you have a high level of data integrity, and so on and so forth, so that you don't have to recall a batch in case there was a failure detected later on. Validation of gaseous sterilization process involves but is not restricted to the following. So, bioburden, manufacturing area environment, determination of time and humidity in preconditioning area, determination of temperature, pressure, time and humidity in the chamber, ventilation of the load after sterilization, loading patterns, biological indicator survivals, and vendor certification of gaseous sterilization treatment is carried out by external contractors. So when you carry out a gaseous sterilization procedure, you need to consider all these elements as a part of your process. Just like we did for gamma irradiation, here also determining the initial bioburden, ensuring that the manufacturing area environment is right. If you're doing it in a grade A in an isolator, you have sealed the isolator properly, there are no leakages. If there is a proper condition which is there, remember that gaseous sterilizations are dependent on humidity of the environment and the time. So what is the humidity which at which you are injecting the hydrogen peroxide, for example? So those are very, very important aspects. Okay. Sporicidal process. This is just a PICS statement. I'm not going to read through it. It's there for you to refer to it. Mechanism of lethality is known to you. It is oxidation of the microbial components. When you select a biocide, you have to worry about material compatibility. You have to make sure it is environmentally safe. It is not going to cause problems of health to the people and is safe to handle. Site production experiments, of course, the place where it is manufactured, you have to audit it and make sure that it is being manufactured with a consistency, and therefore you will get the right quality without any other contamination into the biocide every time you buy from a particular vendor. And then, of course, validate the whole cycle of the biocide itself, or validate the process. Okay. Now, we will take VHP decontamination here as an example for validation, and I will take you through some of the steps which are there. The reason for taking VHP decontamination is the most widely used decontamination agent in the industry today. Dosage rates are between 400 to 1500 ppm. Less than or equal, greater than equal to 5% humidity is where you can start with. Generally, it is around 30% humidity. And when you are doing the hydrogen peroxide vapor injection, it goes, the humidity inside the chamber will go all the way up to 90%. We've seen that through the curves when we discussed VHP decontamination. Uh, Less than equal to 25 degrees centigrade temperature during the whole process, and the cycle time is anywhere between 20 minutes to 1.5 hours. This 20 minutes to 1.5 hours is the dwell time and the startup time where you build up the concentration. Aeration times could vary. Of course, today a lot of isolators and a lot of VHP decontamination cycles are achieved within less than 2 hours or within 2 hours, and therefore these are the typical cycle times. Okay, this is what happens. You have the conditioning, you have the exposure, and then you have the aeration. So the value of the H2O2 goes up, and then it goes up to, in this case it is, the dosage rate is around 800 plus or minus something. Always remember that in any pharmaceutical application, whenever you specify a parameter, temperature, pressure, dosage of H2O2, dosage of gamma irradiation, temperature for sterilization, you always have to specify it as a range. Even the temperature of your room is never a singular value. It is always 23 plus or minus 2 or 24 plus or minus 2. Humidity is always a range. The regulators are looking at the higher and the lower limit defined around the central point that you have determined for everything. So it is never a constant value. If you say, I always maintain my room at 22 degrees centigrade, I do my sterilization at always at 123 degrees centigrade, it will not work. You have to always specify a range, because you will see that today with the advanced level of automation, in earlier days when I used to run manufacturing, we used to manually record data. So we did not have and there was limited staff, so we used to record it once in every 5 minutes, the temperature, and use it for, let's say, 45 minutes. So we had 9 readings and it was fine. Okay, even then we could see a variation that would go up and down. in that situation. Today, with the automation, you have a capability to record the data of the temperature or whatever you are determining, in this case H2O2 ppm, every minute or in a every 10 seconds, 5 seconds. These are settable on the automation system. So when you see the graph, it will always look like this crazy graph going up and down, a sinusoidal wave, which you will see because the way there is a variation within a band. What is important to therefore write in a graph is the band in which you are allowed to operate. So you will have a line here which will say this is my lower tolerance limit, this is my higher tolerance limit, and as long as the sinusoidal curve is between those two lines, you have a proper exposure process in this case. Okay, so that's something that is important to determine and specify and make sure it also appears in the graph. Okay, now validation of a decontamination or a gas sterilization cycle. I'm giving you a list of references where these processes or procedures are available, right from USP to European Pharmacopoeia to the EU GMP, Utralex Volume 4, Annex 1. PICS has got recommendation, ISO has a specification 11139. PDA technical report number 51 and has some data on this. So these are some of the references. Any validation process is a documented program that provides high degree of assurance that a specified process, method, or system will consistently produce a result meeting the predetermined acceptance criteria. So any validation process is a documented system which says that by doing this repeatedly, you will get a consistent end result. Okay, we have gone through this process of equipment qualification in the autoclave, the DQ, URS, DQ, IQ, DQ, FAT, IQ, OQ, PQ. So equipment qualification, I'm not going to spend time on because we have done already talk on this. Very similar processes are followed in each and every equipment which you get in the facility. So if you are doing an isolator where VHP decontamination is happening, or a VHP pass box where you are doing the VHP decontamination cycle, you have to pre-qualify the equipment from a URS, DQ, IQ, OQ, PQ perspective. Okay, without that you cannot start. So I'm not going to go deep dive into that. There are certain ISO definitions which are already provided here for reference, and you can read through them. Okay, this is a very important part of it, which comes as a part of the empty chamber parameter distribution. What do you do is that you take an empty chamber, be it an isolator, be it a VHP pass box, whatever is your enclosure in which the VHP is happening, you will put an indicators which are chemical indicators. Now here you use chemical indicators because these chemical indicators which are there, if there is an exposure, that particular purple dot that you see will turn yellow after the exposure if it has been exposed to the right quantum of H2O2. So what you do is that you place these indicators throughout the chamber, the chemical indicators, run the dose and see that if there is a sufficient exposure of H2O2 within the chamber, because you are doing an empty chamber mapping where you are making sure that your empty chamber is exposed properly to the VHP dosages or the VHP concentration to achieve the right sterilization cycle. So you will place them at different locations. You can see it is placed on the four corners of each side. Then it is placed in the middle. It is placed in the center of the chamber. And then based on this, one will be at the front, one will be at the back, the different positions where they are placed. And then the vapor cycle is run. And these indicators, when they turn the color from this color, which is purple, to yellow, or the indicators could have different colors, that they could turn green, they could turn whatever color, the which is defined by the chemical strip manufacturer. This here is a chem dye which it turns into green. This is another vapor strip which is given by another vendor which turns into yellow. then in such a situation you will be 100% sure that your this thing is very clear. And now this is just an example of how many are kept. You can keep any number on an empty chamber. So you will do this on an empty chamber and make sure that your chemical distribution of H2O2 is right. Then you will do what is called as a load mapping, and we will talk about biological indicators. Load mapping, we have again discussed a bit during autoclave cycle development. We talked about the load patterns, the fixed load, fixed position, fixed load, variable positions, variable loads, variable positions. Generally, in case of VHP, you will not generally have a variable load, variable positions, because then it becomes very, very difficult on a VHP cycle because of the fact that your loads are not distributed. Most likely, you will develop cycles on a fixed load variable position basis. So the same situation that you look at penetration of the H2O2 by putting biological indicators, determine the difficult to sterilize loads, and use that as the effective measure by placing the biological indicators during the routing process. What are biological indicators? We have already seen them in the past. Well characterized preparation of a specific microorganism that is known resistance to a specific sterilization process. We had Bacillus stereothermophilus for autoclaves, Bacillus ectrophius for dry heat sterilization, and so on and so forth. So, in such indicators are what is used. These are your friends. Please remember what you saw in the other one. In the gamma irradiation, these guys cannot be used. So biological indicators are very good friends. So you will do an empty chamber mapping, then you will do a load mapping where you will place the load inside the chamber along with the biological indicators and the chemical indicators. You can place chemical indicators and biological indicators during the initial validation of the cycle and determine whether effectively the H2O2 has penetrated or not. Okay, obviously you will place multiple BIs, which is very, very important. 6 log reduction is what is definitely asked for. You cannot just put one BI, because if you put just one BI or say two BIs, then what is the or just in one BI you place two biological indicators. One became okay, one did not turn purple. So this means it was the cycle passed, which is means that there is a growth here, which means there is a positive here, which is a failure, and there is no growth here in the biological indicator. It remain purple here. So in this situation, which is there, how will you determine whether this location is pass or fail? You can't, right? So you will have multiple placements within the chamber, and within a chamber you will have at least 3 BIs per location, so that you can have an average. So this, for example, is pass, two pass, one fail is also pass, whereas three is complete failure, two failures, one pass is also failure. So this is what is the result that you will look for. Ideally, this is what your target is, but you may end up having these kind of situation. But from a statistical point of view and from a regulatory point of view, this is also considered as pass. So you will place the BI and you will do a process confirmation. The last stage is process confirmation. You will run the actual process with BI indicators and place them initially at multiple locations. You will determine the worst case locations, and then on a routine you will only place on those locations and determine whether your cycle is okay or not. What you will also do is you will do an audit periodically to make sure that you repeat the load mapping with the BIs extensively placed to make sure that the microflora changes have not affected your VHP sterilization cycle process. Okay, so this is how you do the validation. So when I look at it from a validation perspective, it is more or less a situation which is there. Any sterilization process that you see, you have to first determine what is being sterilized. You have to study the properties of that. You have to fix the dosage rate at which you are going to do the sterilization. and then you will do certain lab studies to make sure that data is available. You will fix a range at which it will happen. In case of VHP and autoclave, you will do an empty chamber cycle. In case of gamma irradiation, you can't do an empty chamber cycle, so you will do with a minimal load or a load mapping. You will always do a load mapping for a new facility or a new autoclave. You will use biological indicators, of course, for VHP, heat, sterilization, etc. Only in case of gamma irradiation, you can't do that. So you will put a you will do a bioburden pre and post. And then, based on the biological indicator load mapping, you will place the biological indicators in the worst case scenarios and run the process, and you will keep on running the process consistently at the parameters that you set. Very, very important that each and every parameter which affects your process of sterilization is mapped properly. So with this, I'm opening the session to question and answer, because I think I have taken more time than I was allocated. But let's quickly answer a few questions. Whatever questions you may have, please shoot your questions, and we will see what has to be the next, and we'll move to the next one. So questions?
So can you explain the verification tests?
Verification where?
Sir, in the validation of
You're talking about gamma irradiation or VHP?
gamma radiation, sir.
So in gamma radiation, you're talking about dose audit or routine processing, or you're talking about placement determination. Which one? Joshua, where do you have the doubt?
So the verification test, so the plus or minus 10 degree
This one,
at yes,
those audits.
10%.
Okay. So what you do is that what I told you is that the natural, here in this case, there are no biological indicators, which means that you do not have a biological strip which you can place inside the load and determine whether your gamma irradiation was successful or not. What you are reliant on is the determination of the initial bioburden and the final bioburden, and see whether there was a reduction in that. But the bioburden that you started with was based on maybe you developed this process 1 year back. You 1 year back, your bioburden in the environment in which you were manufacturing this particular product of medical device or a pharmaceutical or any other product, the bioburden would have been different. Why would it have been different? Because we are living on an earth where there is a dynamic change in the environment outside us. If you take a microflora mapping of the place you are sitting today, Biocon Academy, today, and you do the bio study of the flora, say 1 year down the line, you will see that there could be either addition or deletion of a kind of microflora which is present in the environment. Okay. Now, in such a situation where you do not have a biological indicator available here, what you need to do is to do a routine dose audit by checking the natural bioburden which is present in the product uh, at the time that you are doing this audit. And at that point in time, you will go to a research irradiator. You cannot go to a plant scale equipment, correct? So you will use a research irradiator and you will take the verification dose. So now verification dose, what had you taken? You had said I want to use 22 kilograys plus or minus, let's say 5 kilograys. Okay, you determine this as a range for the routine process. Now you will make a tighter tolerance when you do the check, when you are or doing the audit. And if it was 15 sorry 22 kilograms, you will say within plus 10% of that radiation dose or minus 10% of that radiation dose, I will subject the product to that tighter level of radiation dose, and not the wider range which you have, the window that you have specified in the commercial production, and see whether my sterility assurances are being still achieved. Okay, so you will work in a tighter window when you are doing an audit as compared to a wider window which you are allowed during the commercial manufacturing. So verification dose is with the tighter dose tolerance when you are doing a dose audit or when you are determining the dosages also. Clear?
Yes, sir.
Any other questions? Okay. Um, if there are no questions, let us meet in the next 5 minutes, because the next topic, which is biomanufacturing, is a slightly longer topic. So I want a little bit more time to explain that, because it's a topic which we are touching. So far, we've talked about sterile manufacturing and all. Now we're getting into biopharmaceutical manufacturing process, which requires a little bit of more time for us to discuss. And therefore, can I request all of you to be back by 11:20? It is 11:12 right now. 11:20. Do a quick bio break or do a quick tea break, whatever. 11:20. We will start the next session. Okay.
Okay.
All right. So I'll see you at 11:20. Okay, shall we start?
Yes, sir.
Okay. So, welcome back to the last session for today. It's an introduction to biopharmaceutical manufacturing. We've been hearing a lot about biopharmaceutical manufacturing. We hear biopharma, biologics, biosimilars day in and day out. So let's just understand what is the biopharmaceutical manufacturing process. Okay. Uh, okay. So biopharmaceutical manufacturing or biomanufacturing is a specialization within biotechnology. So it has its base with biotechnology. It's an advanced technology manufacturing industry responsible for making biologics. Okay, so it's nothing but a biotech process which generates biopharmaceuticals or biologics. What are the biopharmaceuticals or what is biopharmaceuticals? If we have to say, the process of manufacturing biopharmaceuticals is called as biopharmaceutical manufacturing or biomanufacturing. Biopharmaceuticals in turn are any biotechnology based therapeutics that structurally mimic the component found in the living organism. Okay, so in a living organism there are a lot of components, lot of stuff which goes inside our body, a beautiful machine inside. Biopharmaceutical uh, is a product which, when prepared through the biotechnology route, mimic the component found in the living organism. So, let's take an example of one of the oldest biopharmaceutical which was available, which is insulin. Insulin used for control of diabetes and sugar within the body, sugar levels within the body. Was extracted out of cow, bovine, and from pigs, and it was administered to patients in history till a biotechnology based process was found. And I will go through that history in this presentation to tell you how, when, and what was done. That you can inject it back into into the living being, and it will mimic the same components. Now, what are these components? It could be a hormone, it could be a growth factor, it could be a blood protein, it could be a clotting factor, it could be an enzyme, it could be an antibody, it could be a DNA or an RNA or even a stem cell. So all these categories or components which are found in living organisms are now available as biopharmaceuticals through the process of biomanufacturing. Okay, there are numerous applications in health and medicine for the biopharmaceuticals. So, therapeutic proteins for treatment of diseases. There are therapeutic proteins, insulin is one of them for diabetes, for example. Vaccines to prevent diseases. All of us have taken vaccines, influenza vaccines. We've taken vaccines for rotavirus is given for as a vaccine to animals. Then we have vaccine of COVID, which was taken, which was based on a DNA base. So vaccines are also applications coming out of biopharmaceuticals. Protein or DNA based diagnosis. So, a lot of times to be able to diagnose a disease, you need to use a biopharmaceutical or a protein or a DNA, which is coming out of a biopharmaceutical to determine whether a particular sample is contaminated, not contaminated, is showing certain signs of growth. So, that's an application in diagnostics. So, there is treatment of diseases, there is vaccine to prevent diseases, there is diagnosis. Regenerative medicine technology is a relatively new field here, where you are allowing the regeneration of the medication itself into your body by introducing these components into your body. And gene therapy, a very specific treatment done at a genomic level of the body to make sure that you are cured. Okay, so there is gene therapy. So gene therapy is done for various parts, right? It can be done to cure your diseases. It can be done to cure a certain genetic defect which is there. So this is an application which can happen through biopharmaceuticals. Now, what is the biomanufacturing process? Where did it start? It started with the manufacture of a recombinant human insulin, which was commercially produced and marketed in 1982. So, the biopharmaceuticals are much younger than I am. They were only here in 1982. And though the work of getting to this stage of 1982 began way back in 1970s. And what happened is what is the history that I'm going to give you here. But human insulin, or recombinant human insulin, was the first commercially produced and marketed biopharmaceutical. Today, you see, even today, recombinant human insulin is a very important product or a therapy segment which is still relevant in today's world after so many years, almost 40, 44, 42 years, for a simple reason that it is helping patients with diabetes to fight the diabetes care requirements, and therefore still lead a healthy life on a long term basis. Work began in early 1970s. What did the research scientists do? They developed protocols to construct the DNA vectors. Okay, so what is a DNA vector? A DNA vector is the DNAs are made out of various amino acids and sequences. In these amino acid sequences and proteins, the vector is taken. You take a fragment of DNA. So the DNA is cut into smaller pieces, and then you put it into a circular DNA molecule like what is shown here. A fragment is taken, it is taken, a vector is cut. So you take a DNA vector, you cut it, and then you insert this DNA fragment into it. This becomes a recombinant vector. Now, this recombinant DNA would be inserted into E. coli. So you will take this DNA and push it into an E. coli through a process of transformation. And once this is done, okay, then what will happen is that if one piece of a new DNA included a gene that produced an enzyme that broke down a particular antibiotic, the bacterium containing the introduced gene would be resistant to the antibiotic. So what happens is that when you place this recombinant vector into an E. coli, and if this DNA had a gene which was able to break down a particular antibiotic. Now, the E. coli can grow efficiently in an antibiotic environment itself. What does that allow it to do is that it allows this E. coli to multiply because it has got this particular DNA vector, which is modified DNA vector, which is allowing it to grow into the antibiotic solution. The cells which are without the vector will naturally die because they are in an antibiotic, but the cells which are having the DNA vector will survive and multiply. And that is what is the whole process of how it is done, that the recombinant vector is inserted into a media. Okay, E. coli is one of the microorganisms which are used. There are other microorganisms which are also used. There is something called as a CHO cells today. There are other cells which are also used apart from E. coli to do it. But human insulin, when it was produced the first time and is produced even at a lot of places today, it is using E. coli as the base microorganism. Human insulin gene was inserted into such a DNA vector. So now the human insulin gene was entered into a DNA vector like this, and then alongside with a promoter to drive the expression of the gene encoding the protein that would confer antibiotic resistance. So you put it in a DNA. The insulin gene, which was here, a fragment of DNA was inserted into a larger vector. It was then placed in a media where it would grow or would promote the growth of the E. coli into an antibiotic environment. Now, once this was done, then the bacteria would grow into a selective medium. They would produce high level of human insulin mRNA, which was translated into large quantities of human protein. So once this growth happens, you extract the RNA out of it and isolate the protein, which is your insulin. So insulin is nothing but a protein which you insulin is a protein, right? So insulin is a protein which is used for therapeutic purposes. How do you make it grow? You use a fragmented vector or a recombinant vector, insert it into E. coli, grow it, and then you extract it out, and then you isolate the protein out of it, which is then converted into a biopharmaceutical product. So you have a DNA, you transcripts into the RNA, and then from there you extract the protein. This is how the biology works. So there is a DNA which you started with an E. coli, you remove the RNA out of it, and then you remove the protein out of it. So this is the process which was used. This is the genomic process which is there. I'm not going to spend time on that because that's a lot of research work, bench scale research work, and a program which goes on for multiples of years. And we are not covering the that transformation process into the biopharmaceutical manufacturing. I'll be focusing on the commercial scale process when we are discussing this topic today. Here I'm providing you a list of other biopharmaceuticals and the company which was the innovator, meaning the first company which did this work. Of course, today for all these products which you are listed here on both the tables 1 and 2, there are a lot more manufacturers. So anybody who is not an innovator and is manufacturing today is called as a biosimilar manufacturer because they are doing biosimilar manufacturing. Uh, Biocon group of companies, Biocon Biopharmaceuticals, for example, is one of the largest biosimilar manufacturing company in Asia, if I'm right, or and has also got a very large global footprint. And they are manufacturing a lot of biosimilars. Insulin was the first biosimilar product, which was also manufactured by Biocon. And of course, post that, there were a lot of monoclonal antibodies, which were also produced in Biocon from 2001 onwards. So, human insulin for diabetes care, TPA or tissue plasminogen activator for myocardial infarction. Genentech was the originator. Human trope or human growth hormone, which is meant for deficiencies in human beings. Eli Lilly and Company. Epogen, which is a hormone that builds red blood cells for cure in anemia, was built by Amgen. Energix, hepatitis B vaccine, built by SmithKline Beecher. Then interferon for multiple sclerosis, done by Chiron or Bellex Laboratories. Then Gluceres, type 1 Gaucher's disease, Genzyme. You will see that a lot of the companies which started early on, like Eli Lilly, Genentech, Amgen, have multiple of these products. Even today, they continue to explore biopharmaceuticals as a therapeutic area. Just like today, we see a lot of research going on in various companies in India, including Biocon group of companies. Monoclonal antibodies, Herceptin was started somewhere in 1998. 2004 was an Avastin for colon cancer, Vesitipix, which is for metastatic colorectal cancer, and then there is the Erbutix, which is again for head and neck cancer, Imclone. These are various monoclonal antibodies which are produced. Apart from this, there is parallel use or use of these same drugs for other treatments. For example, trastuzumab, which is being produced by a Biocon group of companies, was also used for treatment to COVID patients during COVID-19 incident or epidemic. So there are usages of the monoclonal antibodies not only on the targeted indication of cancers, but also sometimes there are other parallel applications which are there. We are today, I have a biopharmaceutical which was primarily used for cholesterol treatment or high, treatment of cholesterol. Converting the bad cholesterol to the good cholesterol, but it has also found its application, parallel application in nephrology and ophthalmic treatments. So biopharmaceutical that way is a very specialized field where by using the biopharmaceutical products, you are not only treating the target disease segment, but in some cases, not in all cases, in some cases, you are able to find usage in other therapies requirements also. And this, of course, needs to be tested. A lot of phase studies, clinical phase studies has to be done. But this is just a list. This list is a non-exhaustive list because there is a lot of research going on today. Human insulin itself has gone, other analogs which have come in like Blargin and Dispro, etc. Similarly, for cancer treatment, there are various kind of monoclonal antibodies which are available today. So biomanufacturing is a growing field. This is a typical period of launch or the discovery process where you have the initial discovery, then you have a development, and then you have a launch. And what you can look at is the bottom line. This data is probably about a couple of years old. So you can see the time period that, to just do a discovery where you are trying to do some studies to see whether a biopharmaceutical will work or not. It takes about 6.5 years, and the cost is about 350 million. Then once you have about 5,000 compounds which you will evaluate at this rate, that is your success rate here, and you will probably find a few of them. Of the 5,000 compounds that you evaluate, only five will enter into clinical trials. And in clinical trials, you have phase one, phase two, and phase three studies where you have 20 to 100 healthy volunteers, then 50 to 100 patient volunteers, and 1,000 to 5,000 patient volunteers in each of those phases at a minimum. And you then have to worry about ethnicity and different global locations today. And that itself takes about, say, 770, about $400 million to get to a development stage. And then you file an application. Out of those five that you have taken to clinical trial, probably one can be commercialized, one gets approved. So that requires another spend of commercial manufacturing at 1.5 million, sorry, 80 million dollars. And then if you look at the overall final post-marketing testing required by FDA, then that goes on for about 15 years, and that's a cost of 800 million. So the cost of development of a biopharmaceutical is pretty high. So any development which is happening in biopharmaceutical or any manufacturing that you do at a commercial scale, please remember that it has come through a struggle of an initial of at least 5 to 10,000 compounds which were screened. Then of that, maybe about 1,000 of them, 1 by 1,000 of those evaluated entered clinical trial, and there was one which was successfully filed. Now, in such a situation, it is very important that the process of biopharmaceuticals is run accurately, repeatedly, and consistently so that you can deliver the product of the right quality to the patient all the time. One mistake there, and you could lose all this effort which was put if your file gets disapproved because you did a wrong manufacturing process. So let's understand what is a typical biopharmaceutical manufacturing process. I do not claim, or no literature claims, that this is the only process, but these are the general steps involved in most of the biopharmaceutical manufacturing process. There would be a cell bank or a vial which would contain the initial cell. Remember the E. coli that we started with. That E. coli containing that modified vector is in a vial that is then set into a growth. So the phase which is here from spinner shaped flask to seed bioreactors is called as the inoculum growth. You take the cell bank, you inoculate into a media, you allow it to grow to a certain numbers, then you put it into a larger bioreactor where it grows further before it goes into a production bioreactor. Just to give you an example of, again, insulin manufacturing, for example, if a production bioreactor was a 60,000 liters, you would generally see a seed bioreactor N minus 1 at about 5,000 liters or slightly more. And then you would see the pre-seed bioreactor, which is about, say, 500 liters. So it's a scale of 1 is to 10 generally that we have seen. There are, of course, variations there that sometimes this is a 10 kL, though this is a 60 kL, and so on and so forth. And this would be at a shake flask level or in the lab, which would be 2-liter shake flask into 2 or 3 of these shake flasks. So starts from a small vial, a similar vial to what you saw for your COVID-19 vaccine, or even smaller than that, and it gets into a 60,000 liter fermenter. So there is growth at each stage, getting the media composition right, getting the conditions right at each of these stages, so that there is growth and there is no other contaminant which is growing. Because remember that you are giving a media which is good for growth of all kinds of microorganisms, right? So if this particular microorganism was growing here, and if you did not have the right conditions, then other microorganisms could start growing, and you probably lose the entire growth media which you were targeting. So in that situation, it is very important that at every stage of manufacturing, you take proper care to ensure that your manufacturing process is well controlled, so that there is no contamination coming in, and the product of interest is what is generated. Fermentation, we are not covering in any of the sessions of biomanufacturing, but is an important and a separate topic. I don't know if somebody has already covered it for you through other lectures, but there are various factors when we consider fermentation process in terms of control, in terms of the media sterilization, growth of the microorganism into the media, controlling the parameters like oxygen availability, etc., within the bioreactor, and making sure that you grow the cells of interest. There will be other microorganisms which may be present, but what is important is to grow the cells of interest. So, fermentation by itself is a different subject and a large topic by itself, but this is the production process. So, you go through growth phases of seed bioreactors till you reach a production bioreactor. Then you separate out the cells from the rest of the media through the process of centrifugation. Generally, you send this entire mass into a centrifuge which is rotating at a high speed. G force has to be calculated there. It separates out the solid materials from the liquid materials. Sometimes the product is liquid is of interest, sometimes it is the solid which is of interest, depending on whether the product is intracellular or extracellular, because your product can be remaining within a cell or can be growing extracellular. So if it's extracellular, then generally it is present in the clarified liquid or the liquid part of it, and you need a clearer liquid. Or if it was intercellular, then you will probably end up having the solid, which is of importance. Both of the processes are possible. Then here we are talking about the liquid being of importance. So the product was coming out in the liquid. You further clarified it. You are using a depth filter. Sometimes the centrifuge itself will give you a clear liquid that you don't need to use a depth filter. But in an event, you need to use a depth filter. Centrifugation does the primary clarification, depth filtration does the secondary clarification, and you get a clear liquid which is containing the product of interest. Now you have to isolate the product. So you do what is called as a chromatography step, where you do affinity chromatography. You remember that this was a live cell. It was having E. coli, which is there. So there is also production of virus along with the rest of the mass, because this is nothing but a mammalian cell. There are various viruses which are present within our body itself, but they are good viruses, right? So even in this liquid, there are going to be viruses. You need to inactivate them. So some depending on what kind of product you are producing, generally for all the monoclonal antibodies and most of the therapeutic proteins, you need to deactivate the virus. So you do inactivation of the virus by changing the pH. Then you again clarify it by doing an ion exchange chromatography or a hydrophobic interaction chromatography, or both. Then you do a filtration where the virus gets completely removed. You concentrate the solution by using an ultrafiltration module or a cross flow filtration, and then whatever product you get as a drug substance is ready for filling, meaning it is a bulk drug substance. Now that is then formulated to get into the injections or the formulations which you will use for the individual patient treatments. So that's the whole process of biopharma manufacturing. So there is the fermentation part or the bioreactor part of it, followed by the clarification or primary downstream. Then you have a primary product capture which happens, viral inactivation which happens, fine purification which happens, final viral filtration, concentration, and the final drug substance which is ready. And this drug substance then in turn goes to make your formulation for the drug product. So let's go through that different steps what I told you. So the vial, thaw, and seed in train. So these vials which are there are generally stored at -20, -80 degrees centigrade, because you do not want them to grow wherever they are sitting in the vial itself. So they are stored at a low temperature so that they do not start to grow or get have a death kinetics which is generated there. Remember the death kinetic curve that we study in the sterilization process. So a single vial of cryogenically preserved cells is thought to start with. So it is frozen at -20, -80. You cannot use it as it is. So you need to thaw it in a controlled manner to start the seed train for mammalian cell culture process. Vial thaw can be accomplished at room temperature, or you put it on a heating block, or in an incubator, or in a water bath. So you could just leave the vial outside and it will thaw, just like you take something out of a freezer. If it is already kind of frozen, you need to throw it to thaw it. So if you keep the ice cream at room temperature, it starts to thaw, right? So a similar situation, you could do it on a room temperature. You could do it in a controlled water bath temperature by taking the vial and placing them inside a water bath of a certain temperature so that it thaws. You could use a heating block, which is a dry heating mantle, which is there, in which you can do controlled temperature, or you could just place it in an incubator, which is already at 30 degrees, 37 degrees, or even 25 degrees. It will thaw. Generally, that's how it is done. Now, resuspension process. Now, you take an enriched medium, you give it a lot of growth media, and to dilute any preservatives, because there would be some preservatives added for it to be frozen. So you take that into an enriched medium to dilute them out, and then resuspend the cells into that enriched medium. Then transfer the thawed cells into different volumes to target a specific range of initial cell density. So you prepare media, fermentation media or the growth media into shake flasks or into spinner flasks or even into certain small lab scale reactors, like wave bags or something, and in that you put the cells or transfer the cells aseptically, and you achieve a certain cell density. So you are growing the cells in these media in a shake flask or in a spinner flask or whatever the equipment you use, so that you can achieve a cell density. Without this achieving the cell density, if you try to send it to the seed bioreactor, then what will happen is that it may not be able to grow as fast and in the time and the density that you wanted. So it is important to get the density right at this stage. This is measured through a spectrophotometric analysis or microscopic analysis, or microbiological processes, or sometimes both. So this is to achieve an initial cell density. Once you have achieved that, viability ranges vary with each process. So what happens is that because you are having multiple wires, you may have got wires in one lot from the source, which could be ATCC or any other source, and or working culture that you have got from your R&D department. Now that's available because of the aging of that wire, because of the media composition. You are using different batches of the media. You could have a different cell density which is grown. The process is consistent in terms of reproducibility, but there is a slight variation. Remember the tolerance thing that we spoke about in the sterilization process. Here also you will have a tolerance where you say that density, I cannot use this. So generally, when you look at the stage, which is spinner shake flask or seed bioreactors, there are multiple of these which are running from a single vial bank, so that if one spinner cell shake flask fails to give you the cell density which is required, you could use another one. Okay, because you can inoculate the cell which is in the vial into two or three different flasks, for example, and allow them to grow in those enriched media. It is generally an enriched media, so there is never a dearth of the media for growth which they require. Okay, now the number of stages in a is, it is possible that you could directly go from a wave bag or the single stage into the direct n-1 stage. Sometimes you need two stages, sometimes you need three stages. So this will vary with every molecule and product that you are trying to produce. It may utilize T flask, shake flask, spinners, wave bags, various options which are available. Volume of the culture varies, and this puts demands on the design and footprint of the manufacturing area, because you could be considering multi-product facilities. You can imagine that the culture volumes could vary. Some cases you need 1:100 ratio. Some cases you need 1:50 ratio. So in that situation, understanding the different type of products that you are planning to produce and what are the requirements in terms of cell densities and therefore the volumes becomes important to determine what kind of a manufacturing area you need to put. Generally, what is done is that you take a product which is going to be your largest manufactured product, design the facility onto that basis, and then determine what is the capacity of other products that you can produce in the same facility. For example, if you had a 60,000 liter fermenter which was manufacturing, let's say, product A, and there was a certain output of a product A, using the same fermenter for product B, you might have a lower or a higher output depending on the volume to culture ratios that are required there. Okay, now concentrations have to include a number of containers at each stage, multiple trains running in parallel, equipment needed, utilities, classification of the room, open or closed processing. All these factors have to be taken care of when you are designing a facility. Okay, seed bioreactor used for cell expansion. You take the shake flask, put it inside a seed bioreactor, which has got the right media, right conditions of temperature, aeration, etc., so that you again get to the target cell density for the production bioreactor. Now, seed bioreactor, the factors to be considered is the size and the number, depending on 1 is to 10 scalability, 1 is to 5 scalability, depends on the size. So you need to decide what is the size of the seed bioreactor, what are the number of bioreactors, how many bioreactors will I run to support one production. Sometimes it's you may inoculate two different seed bioreactors into one production bioreactor, or sometimes you run them parallel there so that if one fails, the other one is available, and then you throw the other one down the drain just because you can't inoculate it into the production fermenter. The method of inoculation, are you transferring it in a closed fashion aseptically, or are you going to collect it and transfer it? There are various methods which are used. So generally, of course, today closed transfer methods through aseptic processing techniques is what is the preferred methodology, but that also needs to be taken into consideration. What is the area classification do you need? Do you need a area to be classified as grade D, grade B, grade C, grade C or grade D, or can you do it in a controlled but unclassified areas? A large, very large fermenters are generally run in ventilated areas. If you walk around large manufacturing facilities which has got 100,000 liter fermenters, 60,000 liter fermenters, these are generally not classified areas. Okay, parallel trains only to provide backup if you lost a seed bioreactor. I told about it. Requirement for addition. Now, when you are doing these culture growth, you may need to add nutrients to the bioreactor while the growth is happening, because certain nutrients get exhausted fast, or you may need a certain nutrient after a certain cell density is achieved. So in that scenario, you need to consider what are the additions that I need to do and how you are going to do those additions, because those also have to be sterile. They have to be sterilized before they are inoculated into the seed bioreactor itself. And likewise, when you run a production fermenter, if there are any additions during the growth phase, during the process production phase of the bioreactor, then you need to do these additions in a sterile manner. The components need to be pre-sterilized before they are added into the reactor, because if you add a microbial contamination through those additions, the microorganism will start the new contaminant will start to compete with the product of interest or the cell of interest, and the media is available for both, because remember, you are growing it in a media which has got all the carbohydrates, protein, sugars, vitamins, everything which is there. So any microorganism will grow. If you add a contaminant, it will start growing very fast, and you can lose a production reactor altogether. And the meaning the contents of a production reactor get contaminated and you have to throw it out. You have to have a process control strategy, the pH, the dissolved oxygen, the type of agitation that you use, the temperature at which you are doing, and what is the kind of automation and process monitoring that you're doing at each stage of this growth. So it is less on the wave bioreactors and all, because they are very simple equipments. They are placed in an incubated chamber at a fixed condition of temperature and shaking, etc. And you are then growing the culture, and those are normally short cycles of probably a day, 24 hours, or sometimes even less than 24 hours. In the seed bioreactors, sometimes it is 24 hours or more, and in production reactors, it could be 7 days fermentation cycle, 5 days fermentation cycle, depending on the bioreactor, the product of interest. And during this entire period of production, you need to maintain the pH constant, because as and when more and more microorganisms are produced, they will also have an effect on the pH. They are like human living organisms. They will have secretions, they will have excretions, and those will vary the pH. So you have to keep on making sure that the pH is in the right range, so that the product or the cell of interest is growing in that pH. If the pH varies, the cells could start dying. The oxygen level is right because they need to breathe and grow. The agitation is right. It's not too vigorous that it starts to break the cells. It is gentle enough not to break the cells. At the same time, it is sufficiently there to make sure that proper mass transfer and heat transfer happens. If the agitation is not okay, you could have inefficient mass transfer, meaning although you are pumping in air, you are pumping in oxygen, it does not reach every part of the fermenter if the agitation is not correct, if the mixing is not correct in different terms. Likewise, when you add a additional nutrient into the fermenter, if the agitation is not right and does not get mixed up, there will be pockets within the fermenter where in some places you will have excessive nutrient available. On the other side, it is starved of the nutrient. In the starved region, the cells will start to die, and in the enriched section, it is unused media. So all these are important factors while designing a bioreactor. It is very important to kind of have a scalability from the lab studies all the way to production to make sure that the agitator, the mass transfer coefficients, the heat transfer coefficients are maintained. Temperature is important because body, just like living organisms, the organisms when they grow will produce heat, and this will raise the temperature of the media. You have to control the temperature of the media so that the it does not grow excessive to kill the microorganism. At the same time, it does not grow to a stage where if there are other contaminants present, and they will sometimes be present, they start to grow faster than the product of interest. So temperature control, pH control, dissolved oxygen control, agitation control, very, very important factors at every stage of the growth that we are talking about. Of course, automation and process monitoring helps if you do it. Production bioreactor, again, size matters, process control strategies are there. Generally, on production bioreactors, you don't have parallel trains. You could be running each bioreactor as a separate process and call it as a batch. Parallel trains are very rarely used at production scale or at a production bioreactor scale. Area classification, what is the kind of classification in which you are placing the area, whether it is grade D, grade C, you have to, it depends on the type of process you are running, and again, requirement of additions because you need to add it aseptically. This is a picture of a bioreactor which is there. It's a large one. I think this is about 10,000 liters. You can see the amount of piping which is going in and out of this. Some of it will be used for sending air, some of it will be used for getting out air of the system. There are pipes which are bringing the additions or the nutrients into the system. Okay. Now, production bioreactors, you also need to understand the harvest criteria. What is the viability of the harvest? Are you sending it to a centrifuge, depth filter, etc.? What is the duration in which you are going to complete the process? So, when do you stop the process of fermentation? Is it 5 days, 2 days, 1 day, etc.? And what is the growth factor, the cell density you are targeting? Is it 1 gram per liter, 2 gram per liter at the end of the day, that of the cell that you're talking about, and even more, is something that you have to determine to say when will you start the production bioreactor, when will you end the production bioreactor. Automation and process monitoring could be based on glucose monitoring, lactate monitoring, or the carbon dioxide coming out of the system, because again, they are living organisms. They breathe, they take in oxygen, they release carbon dioxide. So you can do a growth analysis based on glucose levels monitoring that is depleting in the media, or the carbon dioxide level which is being emitted out of the media. Okay. Now we come to the harvest or the clarification, referred to as harvest because you are taking the fermenter and clarifying it. So it's called as a harvesting of a fermenter, a term that you will come across in biomanufacturing very often. What is your harvesting cycle? That's what people will ask you as a question. And harvesting is nothing but the separation of the cells from the cellular debris and from the harvest growth. Okay. So method is centrifugation, depth filtration, microfiltration, or a combination of these. It could be centrifugation and depth filtration. It could be microfiltration and centrifugation. There are various permutation combinations of the methods. Microfiltration is nothing but tangential flow filtration that you will use using membranes, either hollow fiber, spiral wound, ceramic, whatever. Centrifugation is the process that I'm showing you there. It's generally a disc stack centrifuge where the solid and the liquid get separated. You send the liquid into the centrifuge through one of the inlet nozzles here. Then what happens is that it goes into the dispatch, which is rotating at a very high speed. The solids go and separate out through centrifugal force. They get separated out and they go to the end of the disc stack, and then they get removed into a solid discharge port, and then the liquid, which is the clarified liquid, comes out as a from the other port, because it is this stack has got holes for inlet and outlet. So that is how a disc stack centrifuge works. There are other kinds of centrifuges or separation equipment also used, which are just spinners, but at a large scale, disc stack is the most widely used centrifuge. Area classification, redundancy in terms of equipment. Can you do with just one centrifuge? Do you need multiple centrifuges? Just because if the centrifuge fails, your harvest has to happen once in 7 days. If the centrifuge fails at that time, then sometimes you need a additional centrifuge. These are robust equipments. Of course, you don't need to really worry about getting them kind of having redundancy all the time. Also, because these are to be opened, cleaned, sterilized at times. So in that situation, these equipment become expensive as well. So you may not be able to afford redundancy of every equipment. You can't have a backup for every equipment. So you redundancy is something that you decide based on the criticality and affordability. Automation, of course, is a part and parcel of it. The chromatography, there is a number of steps involved. You need to decide the kind of resin which will be used for chromatographic separation. Okay, and then what are the steps involved in chromatography? What are the loading conditions? What is the flow rate at which you are going to send the liquid into it? At what pH? At what conditions of temperature are you going to send it? Because in what is happening in chromatography is that there is a affinity which is happening, or an ion exchange which is happening, and in both cases you need to determine the right conditions at which the affinity or the loading or the ion exchange will happen. What are the volumes that you will do? Because what is the process? You absorb the material onto the resin, and then when then you pass the liquid so that the product of interest gets out into the liquid stream. So what is the volume that you need? How are you going to measure those volumes? Whether it's a single cycle or a multiple cycle. Can you use the resin only once, or can you reuse it by washing it out? All because there is a cost consideration to it. What are the buffers that you are going to use? Because you will use a buffer for first regenerating. You will regenerate the resin. You will equilibrate the resin so that the condition within the resin bed are at the same pH, at the same ion exchange levels before you send in the product of interest. Then after you have sent the product of interest, you need to remove the product of interest by desopping it. So what kind of buffers you are going to use also becomes important, because you are going to collect fractions from this, and those fractions are where your product is. If you do not use the right kind of a buffer, you could lose the product with certain impurities which are also coming out of the system. Okay. So the size becomes important, sample becomes important, temperature control, mixing of the liquid when it is sent into the chromatography process becomes important. What are the manipulations that you need to do in terms of pressure, temperature conditions, flow rate conditions, and whether you need to do a filter before you send the liquid into the chromatography or post processing. These are all important factors which determine the type of chromatography system and the train around it. The area classification, processing temperature, redundancy of equipment, automation are all common factors which you will see through each and every process step. Tangential flow filtration, here what you are doing is that you are going to concentrate the volume so that you end up with a smaller volume which you have. You can't be having too dilute of product, because then when you do the formulation for injectable it won't work. So you have to concentrate it, the protein quantity to the protein or hormone or whatever is your biopharmaceutical, you need to concentrate it sufficiently large to a volume where it is then usable for further processing. What are the factors? What is the membrane MOC? What is the initial volume? What is the final volume? Are you concentrating at 10x? Are you concentrating at 5x? What is the time which is required? And therefore, what is the processing area of the membrane that you need? What are the operating conditions in terms of transmembrane pressure, temperature, etc., and what is the amount of final product that you will get out, and the system hold up which is there, because this again will have liquid into it. Will you use a buffer to kind of drive out the product of interest, for example, or will you do a certain amount of water addition to drive the product and obtain your concentrated mass, which is sticking onto the membranes? These are all important aspects to be considered during tangential flow filtration. Whether you will do buffer exchange, etc. These are, so this is how a commercial system looks. The earlier photograph was a old photograph, so I picked out the photographs of a modern system. This I think are Sartorius systems. Yes, they are Sartorius systems. So this is somewhere where your membranes will be sitting. The rest of it is the pumping module and measuring the transmembrane pressure and all that, and the control console here, automation. Virus filtration and inactivation. Inactivation is generally achieved by changing the pH of the solution, and then you need to filter it out through a filter which looks like this. So it's a dead end filter, or it's just a single filter cartridge through which you send it. So the liquid is sent out and the clear liquid is collected. So the virus is retained onto the filter and the clear liquid is collected out. So there, what is the membrane surface area which you need based on the volume that you are processing? What is the processing time? What is the load of the viral inactivated virus that you are expecting here? What are the physical changes which the filter material compatibility? What is the filter material, whether it is PVDF, PTFE, cellulose, what it is becomes important. And then physical room change during the process. Are you transferring it from one room to the other? Remember that if you have a virus which is there, where the virus is present, the amount of care that you need to take in terms of environmental monitoring and in terms of personnel protection becomes very high. So in such a situation, you need to make sure that you contain the virus within a smaller processing area and not expose it to many large areas, because in case of any contamination, the decontamination process and the time taken will be large. You do inactivation to pH adjustment, as I said, where you are when you do a pH change, you have to worry about whether the product will degrade at that pH. What is the duration of hold? What is the strength of acid or base used? What is the type of acid and base used? And what is the rate at which you are adding it? Okay. There is also an inactivation by detergent or a surfactant, which is possible, generally not widely used, but it's also something that is a way of inactivating a virus. What a detergent is doing is ultimately pH change only, but it's a way of changing the surface properties so that the affinity also changes here. Not a widely used process, as I said. pH adjustment is what is used here. Area classification and all is fine. The final one is, of course, you have now got the drug substance which is virally activated, filtered, ready. You will fill it in bags or you will fill it in containers. Those containers are then transferred to filling lines. And in a filling line, what happens is you know that you have to do a formulation, you have to do an aseptic filling. Why? Because you cannot sterilize the biopharmaceutical. If you try to sterilize a biopharmaceutical, you will denature the protein or the product of interest. So this is necessarily a biopharmaceutical fill finish operation is necessarily an aseptic fill finish process. You may sometimes need to lyophilize the product, and then of course you have the inspection and all. This is talking about a vial. This line is a vial line from Bosch, but you could be filling it into cartridges, you could be filling it into pre-filled syringes, or you could be doing you could be using a syringe, you could be using a cartridge, or you could be using a vial. In all the cases, the filling operations remain very similar, that either you use a ready to use pre-filled syringes, or you subject the pre-filled syringes or cartridges which are already siliconized, or you may even inline siliconize it. So you will wash them, you will siliconize them, you will wash them again, you will depyrogenate them, you will do the filling. Then if there's a lyophilization process involved, you will send it to a lyophilizer and then take it out of the lyophilizer and do the full capping and sealing, and then you will take it out. If it was a liquid product, you will not go to a lyophilizer, you will seal it at the filling line, you will cap it and seal it with an aluminum cap, and just take it out of the system. So you will do the fill finish. The fill finish here will always necessarily happen in a grade A, but when you are doing a drug substance, you could be doing it in a class A, or if there is a filtration step involved during the formulation, you can do it in a lower class as well. So whether it's a single product use, multi-product use, scheduling, room turnover, what are the shared equipment, what are the shared personnel? Because remember that you're doing it. So generally, if you are building or manufacturing hormones by a biopharmaceutical route, you are not allowed to even share personnel, because personnel can get affected by the hormones which are being processed, right? So in certain facilities, ladies are not allowed to work because you are dealing with hormones which are specific to females, and likewise for the males also. So and they could have detrimental effect on the person itself. A hormone facility is generally not combined with the other therapeutic applications which are there. For example, when you manufacture insulin, you will not produce a hormone. Okay, so these are rules which are given by the regulation, saying that what kind of products can you mix in a multi-product facility and what kind of products you cannot mix, what kind of risk assessments you have to do, what equipments are shared, what personnel are shared, that also gets defined in a multi-product use. Temperature, product stability, operational conditions, we've gone through those requirements already. So this is your whole process from start of a vial all the way to the final injection which comes out, which is administered to a patient or is consumed by a candidate as a prevention, for example, vaccines. So with that, I come to the end of the biomanufacturing process. Okay, not bad. I have still 15 minutes for question and answer. So please uh, shoot questions, and I will go to the slide and explain it again if there is a necessity to do so. So open to question and answers. No questions? Okay. If there are no questions, let us end the session for the day. Thank you for your time and attention to the lectures delivered today. Tomorrow we will meet again from 9 o'clock. We are going to cover three topics: introduction to formulation and mixing technology. We are going to talk about filter sterilization, types of filter, filter selection, filter train, the last kind of a sterilization process that we did not cover so far, a little bit more depth into it. And the most important is the last lecture from a perspective that it covers three large topics. There is filter integrity testing, there is media fills, and then aseptic techniques and behaviors. Now, what I'm going to try and do is to cover at least the filter integrity testing along with the filter sterilization topic, so that they are A, because they are related, and B, the time allocated for the last lecture, which has three large topics, may not be sufficient. So when we do L134, I'll also bring in L135 into the play and try and cover the filter integrity if the time permits. If the time does not permit, however, we will take it as we go along. So those will be the three lectures for tomorrow. Thank you very much for your time, and see you tomorrow morning.
Thank you, sir.
Thank you,
Thank
sir.
you.
Thank you,
Thank you,
sir.
sir.
Thank you, sir.
Thank you.